Tony Cheng from the University of Pittsburgh.
Okay, thank you, everyone. Let me put on my slides. Okay. Okay, good afternoon, everyone. My name is Tony. I'm a PhD student at the University of Pittsburgh in Dr. Najab Owen's lab. Today, I will present our findings on the therapeutic potential of MS-275 in reducing liver injury, inflammation, and fibrosis in a mouse model of cholestatic liver disease. Before delving into the topic, let me give you a little bit of background. Cholestatic liver disease refers to a group of disorders marked by impaired bile flow, which leads to the accumulation of toxic bile acids and triggers inflammation in the liver. In industrialized countries, including the US, the prevalence is increasing over time. Cholestatic liver disease can be roughly classified into extrahepatic and intrahepatic causes. With the recognition of these diseases, we have more patients than in the previous. However, the treatment strategy has been stagnant in recent decades. To that end, developing new therapeutic alternatives is urgent for this unmet need. Transdifferentiation refers to the direct conversion of one differentiated cell type into another without transitioning to a pluripotent stem cell state. Researchers are using it to leverage the strengths to achieve tissue regeneration after injury, including cholestatic liver disease. And preliminary study in zebrafish has demonstrated that MS-275, a selective HDAC inhibitor, can facilitate hepatocyte to cholangiocyte transdifferentiation. HDAC inhibitors were widely studied in the field of oncology, but were seldomly mentioned in hepatology. Therefore, we want to ask that if the HDAC inhibitor has a similar effect on the mammals as in the zebrafish. Our research aims to investigate the efficacy and the mechanism of MS-275 in a well-established mouse model of cholestatic liver disease. We used the DDC diet injury, which is a xenobiotic causing pigmented porphyrin plaques in the small bile ducts and cholestasis. So first, our mice were injected with AAV8 TBG-CRE to label hepatocytes with EYFP for lineage tracing. Then, mice were divided into 4 groups: a baseline child diet negative control, a DDC diet group with DMSO as a positive injury control, and DDC groups treated with 10 milligram or 20 milligram per kilogram MS-275 for 1 week. The livers and serum were collected for downstream analysis. By serum markers, we found that DDC-induced hepatobiliary injury, which was significantly decreased by 20 milligram MS-275. IHC for CD45 showed fewer inflammatory cell infiltration after 20 milligram MS-275 compared to DDC alone. Inflammatory cytokines such as interleukin-1 beta and TNF-alpha were decreased after MS-275 treatment. Using the F480 IHC, we can visualize that macrophage activation from the liver sinusoid in the DDC group, which can be partially reversed by MS-275 treatment. Fibrosis marker genes were all decreased after MS-275 compared to DDC alone. And the polarized images also show that collagen deposition was increased in the DDC alone group, while decreased in the MS-275 treated groups. However, we could hardly find hepatocyte-derived cholangiocyte in the bile duct by the colocalization of EYFP and SOX9, which is a biliary marker in the MS275 treated livers. Although we did see some dual fluorescence positive hepatocytes present in DDC injury along livers. On the other hand, early biliary marker genes EpiCan and SOX9 were decreased after MS275 treatment, while mature biliary marker CK19 was equivalent between injury along and MS275 treated groups, suggesting that MS275 treated livers have more fully differentiated cholangiocyte than DDC alone. Overall, we did not see more ductular proliferation or transdifferentiation presenting in the MS275 treatment group. To confirm MS275's efficacy in mice, we ground liver and did Western blot targeting the histone acylated sites, and the results suggested that MS275 changed the histone acylation both in the promoter and enhancer regions. To further elucidate the mechanism, we sent the liver samples for RNA-seq. Cluster analysis showed DDC with MS275 samples were closer to the baseline than DDC alone. We also found that abundant cell cycle genes were upregulated in the DDC alone group, but downregulated in the baseline and MS275 treated groups. Gene set arrangement analysis predicted several interesting pathways between injury and MS275 treatment groups. Biosecretion, oxidative phosphorylation, and drug metabolism were upregulated in the MS275 treated groups, while cell cycle, senescence, and p53 pathways were downregulated. And these pathway changes were also seen across different databases. We used a colorimetric assay to quantify the total bile acid in the liver and found that bile acids were increased in the DDC diet and decreased in the MS275 group. To further understand the bile acid composition, we employed mass spectrometry to determine the hydrophobicity of bile acid. However, there is no significant difference between the DDC alone and MS275 treatment group. We also use qPCR to validate several target genes, including the genes altered in the zebrafish model after HDAC inhibition. While in our mouse model, these genes did not show significant changes between groups, we found G1S cyclin D1, and cyclin E1 were increased after injury and decreased after MS275 treatment. On the other hand, CYP enzymes involved in lipid metabolism and bile acid synthesis and redox reactions were increased in the MS275 groups compared to DDC alone group. Mitochondrial electron transport chain protein, bile acid transporter, autophagy protein, and bile acid sensing nuclear receptors were all increased in MS275 treated livers. We did IHC for the cell proliferative marker Ki67 and the cell senescence marker P21 and found increased expression around the periportal zone after injury and decreased expression after MS275 treatment. IHC staining also showed MS275 treatment can rescue CYP2E1 distribution, reduce p53 expression, and 4-hydroxynonenal production, suggesting that MS275 restores hepatocyte metabolic enzymes, maintains genome stability, and reduces lipid peroxidation. Overall, the IHC data echo our previous RNA-seq and gene expression assay findings. In conclusion, MS275 shows hepatoprotective effects on DDC injured mice and causes phenotypic changes like improving hepatitis, cholestasis, and liver fibrosis. Through lineage tracing and gene expression studies, we understand that this mouse model can be different from the zebrafish model. MS275 induces cell cycle quiescence in mouse liver, by downregulating genes and pathways relevant to cell cycle, proliferation, and senescence. The compound also activates metabolic enzyme genes and redox balance and affects proteins that maintain genome stability. Further studies, including epigenetic studies, are warranted to elucidate more detailed mechanisms. In the end, I want to thank everyone who supports this project and thank you all for your attention. Thank you.
Great talk, Tony. Do we have any questions? I have one question. So which HDACs is this drug targeting? And then also, do you expect that, or do you think that these effects that you're seeing in terms of protection, do you think it's specific to certain HDACs, or is it just kind of a general property of HDAC inhibition?
Yeah, that's a great question. It's a class 1 HDAC inhibitor, so it's mainly targeting on HDAC 1 and 3, and some effect on HDAC 2. But we haven't tested other HDAC inhibitors in this mouse model yet. So I cannot give you a certain answer, but I believe there's several HDAC inhibitor we can choose, especially on those like already marketing drugs, and also some like preclinical drugs we can test it in the future. Yeah.
If there are no other questions from the chat, maybe I have one. You've seen a hepatoprotective effect both in the zebrafish model and in mice, but the mechanism seems to be different. Have you looked at why this could be? Right, in the fish, it seems to be transdifferentiation, not so in the mouse. Is there something about the fish biology that is particularly different, something that you could explore there?
Yeah, that's an interesting questions, and this is our hypothesis because we think the architecture of liver is different between the fish and the mammals, because in the zebrafish they don't have like zonation, so we guess it's a very different cell-cell interaction and microenvironment from the mammals. So we think there's probably other things going on in the mammals, and we are currently trying to elucidate the mechanisms. Yeah.
Are there any questions from the audience? If there are no more questions, maybe we can move on to the next speaker then. Yeah. So the second speaker today is Brooke Hutchinson from the University of Pittsburgh.
Hello. Let me just share my screen. Okay, wait, this is 1 second. Okay. Okay, can you guys see it? Okay. Hello and good afternoon. My name is Brooke Hutchison, and I'm an undergraduate researcher at the University of Pittsburgh and in the Dr. Najak Bowen lab. So today I'm going to talk about how gut dysbiosis during cholestatic liver disease drives progressive bone loss. And our work focuses on the critical but often overlooked gut-liver bone axis. Okay, to understand why this is important, we first have to look at the clinical problem. We focused on primary sclerosing cholangitis, or PSC, which is a progressive cholestatic liver disease where the bile ducts become inflamed and scarred. A major and debilitating part of PSC is bone disease, specifically osteoporosis, which affects a staggering 10 to 60% of these patients. As a result, these patients have a 23-fold increased risk of fractures. This dramatically reduces their quality of life. However, the mechanism linking the liver disease to bone loss has remained poorly understood, which is what our research aims to fill. So, to understand bone loss, we first need to understand bone remodeling. Bone is a very dynamic tissue which is constantly being broken down by cells called osteoclasts and also being built back up by cells called osteoblasts. So, in a healthy state, these processes are very balanced. However, when this balance is disrupted, which is either by too much bone restoration or too little bone formation, we get a net bone loss, as you can see in this bone loss part. So our goal was to find out how liver disease tips this balance into that bone loss way. So the question is, what connects the gut and liver to the bone? We look at the gut-liver bone axis. So PSC is strongly associated with gut inflammation and microbial dysbiosis. In fact, 70% of PSC patients also have inflammatory bowel disease. So this dysbiosis is known to alter host bile acid metabolism. Also, clinical data has shown a direct correlation that patients with cholestasis have high serum bile acids and low bone mass. So the question is this just a correlation or is it causation? Our recent publication in JCI Insight provided a key piece of this. It showed that disrupting gut bacteria with antibiotics impairs bone formation by interfering with a key gut-liver signaling pathway involving the farnesoid X receptor, or FXR, and a hormone called FGF15. This introduced bile acids as novel mediators in this gut-bone communication. This led us to our central hypothesis. Gut dysbiosis during cholestasis alters bile acid signaling, which in turn drives bone loss. So, to test this, we used a mouse model of chronic cholestasis during a compound called DDC. The DDC diet causes bile duct blockage, leading to hepatobiliary injury, inflammation, and fibrosis, which is effectively mimicking human PSC. We treated these mice from 1 to 11 to 15 weeks of age and then analyzed their bones, gut, and bile acids. First, let's look at the bone structure. So we used micro-TC scanning and we found a dramatic decrease in bone mass in the DDC treated mice compared to the healthy controls. Quantifying this, we also found a significant reduction in trabecular bone volume, which is the amount of spongy bone. We also saw a decrease in cortical bone mineral density, which is the hard outer shell of the bone. So we can see that here the DDC diet causes severe bone loss. Next, we want to know why this bone loss is happening. By staining bone sections, we counted the bone-forming and bone reabsorbing cells. We found that DDC treatment led to a significant decrease in osteoblasts, which are the bone-building cells. On the other hand, we saw an increase in osteoclasts, which are the bone reabsorbing cells. So this is kind of like a double hit where there's suppressed formation and enhanced reabsorption, which explains the rapid bone loss that these patients face. So now we have to go back to the gut. We sequenced the gut bacteria from these mice. We looked at the alpha diversity, which is the diversity of species within a single sample. You can clearly see a decreasing trend in the DDC groups. We also looked at the beta diversity, which measures how different the microbial communities are between groups. Here you can see a clear separation that shows the overall gut bacteria community in the DDC mice is significantly different than the controls. This confirms that cholestasis induces gut dysbiosis. With a dysbiotic gut and damaged liver, we expected bile acid levels to be disrupted. Our analysis of serum bile acids confirmed this. The DDC-treated mice had a massive increase in total serum bile acids, including primary, secondary, and conjugated types. These bile acids are spilling over into the systemic circulation and affecting the bone marrow. Crucially, we also found that these bile acids aren't just circulating, but accumulating in the bone marrow environment itself. This means that the bone cells are being basically bathed in an abnormal bile acid mixture. To prove that these bile acids directly affect these bone cells, we performed a few key experiments. We took bone marrow from our mice and used it to treat bone cell cultures. First, the alizarin red assay shows mineral deposition by osteoblasts. The DDC-treated marrow severely suppressed this mineralization. Second, the TRAP assay shows osteoclast formation. The DDC-treated marrow potentially enhanced these osteoclastogenesis. You can clearly also see that there is no difference in the negative versus the control. This supports the premise of normally there being no effect of bile acids on bone when you are in a healthy individual. This provides direct evidence that the factors in the cholestatic bone marrow are sufficient enough to disrupt bone cell function. So let's bring it all back together in an integrated model. Our data supports this pathway. Cholestatic liver disease and gut inflammation cause dysbiosis. This dysbiosis dysregulates bile acid metabolism, leading to a buildup of specific bile acids in the system. These bile acids then accumulate in the bone marrow, where they directly suppress osteoblast function and enhance osteoclast activity, ultimately leading to net bone loss. So we have identified these bile acids as novel mediators linking liver disease to skeletal complications. I would like to thank everybody in my lab. None of this work would have been possible without everyone here. Thank you.
Thank you for that. That was a great presentation and a lot of work that has got into that. Are there any questions from the chat?
I do have a question. Brooke, very good job. That was super, super interesting. I was wondering if all of that damage could be reversed. Say, for example, if you have the mice on that DDC diet and you take away that diet, give them something like a normal standard diet, or if there's a way to target kind of, we were saying that there was a decrease in the osteoblasts versus an increase in the osteoclasts. Could that somehow be brought back to like an equilibrium state in some way?
So in order for that, I mean, we haven't done any tests with that, but I would assume that you would need to not just stop the DDC diet, but also include treatment. So one of these treatments could be an IBAT inhibitor, which would suppress bile acids from entering the bone marrow. So that's something that I think you would probably have to do in order to get the homeostasis of the bones back to regular. But we have not done any research on that, so I can't say for certain.
Thank you.
So I have a question as well. So I was just wondering about the effects of any possible effects of DDC on the bone itself. Like, is there any, so this is kind of just more about the model. Is there anything you can kind of do to make sure or rule out any like off-target effects of the DDC on the bone and make sure everything's kind of coming more so from the liver?
For that, it's a little hard to explain, I would say, but mostly there's like a lot of other like kind of diets that you could choose. And like you can choose obviously like there's many, but we chose specifically DDC because it's known first to be a really good model for reproduction and of like these experiments, and because it is known to, like I said, give the effects that mimic PSC. I'm not sure exactly if there's another model out there that would be better and like have specifically like no impact on the bone. But DDC is a very, very common diet used to mimic this kind of thing. So I'm assuming that that's like the best model to make sure that nothing else is impacting the bone. It's only the diet which is impacting like the liver and the gut, which then impacts the bone.
Yeah, awesome work, Brooke. I'm always inspired by seeing people doing presenting during undergrad. So, yeah, good job. So,
Question. Do we
oh
have
yeah,
time?
yeah, of course.
That was awesome, Brooke. I noticed in one of your slides you had a paper and it was with Chad Novus. And he taught me a lecture at MUSC and it was about teeth in the oral microbiome with bone. So I was wondering if you looked at any other bones and or teeth and if you noticed anything in your animal model with that.
We only really did look at the bones that I like showed in the presentation, but looking at teeth is actually something that we have talked about in the lab at looking at, just because we've all kind of been a little interested in it. So hopefully we could continue and see like kind of do like another experiment similar to this and see if teeth do get impacted. Um, but so far we have not looked at anything like that.
Cool. Thanks.
I have a question. Can I ask one? Fantastic presentation. So my question is, have you looked at the age and the gender of the mice when you do these diet to see if they correlate? Because bone density also changes with age, and also males versus females, there is disparity in the bone density. So
I know that we did look at age with treating them to 11 versus 15 weeks. But for most of that, we kind of saw very similar results. But we like to look more into that. We would probably just treat them for longer and wait until they're older and see if there ends up being like a different kind of um, results for that. But we do also still obviously like track what age we did extract the bone from them. So we do know like which ones is which.
and how about the gender? Did you see any did you
I don't know for that one.
thank you.
Yep.
Did you have a question, Carlos?
Yeah, so I work in IBD, which, as you said, very highly correlated to PSC. And we see that the fibroblasts not only lose the ability to support the epithelium, they also become inflammatory. I was wondering, since you've seen the osteoblasts have reduced, let's say, osteogenic activity, have you seen if they become more inflammatory? Maybe they're recruiting more of those osteoclasts. Have you looked at all at what happens to them?
We haven't exactly looked at what happened to them. That would be something that we would look for in further research. But like the most of like the inflammation stuff was kind of just about gut inflammation, which then like we know came from because like there were so many bile acids being produced and they were being secreted from the bile acids from the gut. That's kind of like what that kind of inflammation we looked at, but it would be very interesting to look at and see that about like everything else.
Fantastic work. Yeah.
All right. If no other questions, we are doing right on time. So this is great. Keep all the questions coming. The next speaker we have is Ramsey Roner from Tulane University.
Hi. Let me just share. Okay. Hi, my name is Ramsey Roner. I'm a graduate student at Tulane School of Medicine, and today I'm going to be talking about the role of DEC as a cell death switch molecule in liver fibrosis. So a little bit of background about DEC and chronic liver disease. So DEC is an intracellular proto-oncogene made of 375 amino acids. It's found mostly in the nucleus, and its main function is chromatin compaction. A little bit of background about CLD. So we have our normal liver. There's some toxic insult. This can lead to steatosis or inflammation, which can lead to fibrosis. If the insult isn't removed, this can lead to cirrhosis, which eventually death or hepatocellular carcinoma. So the role of DEC in liver disease development is unknown. So I want to examine the impact of DEC deletion in preclinical models of liver fibrosis. So first we obtain these DEC full body knockout mice and DEC plus mice. I'll be calling DEC wild type mice. We injected either a vehicle or a CCl4. Carbon tetrachloride is just a standard hepatotoxin. We injected at 0.7 microliters per gram of body weight intraperitoneal twice weekly for 2 weeks. At the end of 2 weeks, we then harvested their liver and serum and did more analysis. So if we go over to figure B, I did a Western blot to confirm that DEC is fully knocked out, which is what we see. And then we measured the serum ALT AST. So in our DEC wild type vehicle and our DEC knockout vehicle group, we see little to no change in ALT AST as expected. In the DEC wild type CCl4 group, we see an increase in ALT AST. This was kind of as expected. But in the DEC knockout CCl4 group, we see an exacerbation of the ALT AST, a significant increase, which was not as expected, which is very interesting. So then we went and we wanted to see what the fibrosis looks like. And by doing this, we did a serious red stain, which stains for collagen fibers. So in our DEC wild type group in figure D, we see no collagen deposition. In the DEC wild type CCl4 group, we do start to see some more collagen deposition. But it's in the DEC knockout CCl4 group, we see a lot more of this collagen deposition shown by a lot more red staining, suggesting that the fibrosis may be worse. So from here, we wanted to confirm that the fibrosis is worse by, we did an IHC for alpha smooth muscle actin, which is a biomarker for activated myofibroblast. So in the DEC, in figure A, in the DEC wild type CCl4 group, we see little to no stains for alpha smooth muscle actin. In the DEC knockout CCl4 group, we see a lot more staining of this alpha smooth muscle actin. We even quantified this to see that there's a significant increase in the signal of alpha smooth muscle actin in the DEC knockout CCl4 group. We went even further by looking at hydroxyproline, which is a key protein required for the formation of collagen. And we see a significant increase in the DEC knockout CCl4 group. And then we went and looked at, we did some qPCR to look at these 5 fibrotic genes to see if they're upregulated or downregulated or what the effect is for the DEC knockout CCl4 group. And we find that most of them on average are upregulated, 2 significantly are upregulated for the DEC knockout CCl4 group. So from here, we've concluded that when DEC is deleted whole body and treated with hepatotoxin, there is more liver injury, more fibrosis. So the next question we had was we wanted to see if this is hepatocyte specific. So from here, that leads us to our next set of experiments using the AAV8 TBG CRE model. So AAV8 stands for adeno-associated virus serotype 8, which is which just serves as our viral vector that has a high tropism for the liver. The TBG stands for thyroxine binding globulin promoter, which thyroxine binding globulin is a protein almost exclusively produced by the liver, so only exclusively produced only by hepatocytes. So hepatocytes should be the ones that have the appropriate transcription factors to transcribe this CRE gene. This CRE stands for this CRE recombinase that goes in and cuts these 2 LOX-P sites flanking DEC. So in total, this creates our full hepatocyte-specific DEC knockout mice. We injected this virus at 1 × 10^11 genome copies retroorbitably. And then we waited 2 weeks. 2 weeks is our standard amount of time to allow for the CRE to go in and fully cut out DEC specific to the hepatocytes. From here, we then injected CCL4 for 2 weeks, similar to the last experiment. And at the end of the 2 weeks, we harvested the liver and serum. So if we go to figure B, we did a Western blot to make sure that DEC is at least knocked down, which is what we see when treated with the AAV8 group compared to the non-treated AAV8 group. So we want to go even further to make sure that it is knocked out. We did a immunofluorescent stain. The red stain refers to the stain for DEC. Blue is our DAPI, which stands for the nucleus. So when we have this purple overlap, that means there's DEC in the nucleus of these cells, which is what we see in the DEC flox mice, which is as expected. And then if we go down to the DEC flox plus AAV8 in the liver, we see a lot more just blue cells. This is lacking the DEC, suggesting that it is at least knocking it down. We do still see some purple cells suggesting that there is still DEC present in the liver. And these could be non-hepatocytes, or it could be just a few hepatocytes that didn't fully get the virus. But nevertheless, wholesale, it is completely, it knocked down at least. Then we did immunofluorescent staining for the kidney to make sure that this isn't just happening in the liver. And in both cases, DEC flox and DEC flox AAV8, we do see a large amount of this purple, suggesting that DEC is still present in the liver and the kidney in both samples. So from here, then we wanted to look at, let's look at the fibrosis, see if we have similar results, which leads us to figure D. In the no AAV8, no CCL4, we see no fibrosis or no collagen deposition. AAV8, no CCL4, again, a little collagen deposition, but pretty much none. And the no AAV8 with CCL4, we start to see this more collagen deposition. But we see the stark difference with the AAV8 and CCL4. We see a lot more of this collagen deposition. So far, this is suggesting we're seeing similar results comparable to this whole body knockout. So then we went even further, doing some more histology staining. We did our H&E stain to look at our tissue and cellular morphology. In both of the groups not treated with CCL4, we see very comparable tissue and cellular morphology. But it's when we get to the no AAV8 with CCL4, we start to see more of this potentially steatosis. We can even outline a little bit of this fibrotic bands happening. But clearly there is a stark difference compared to the two groups prior. As well with the AAV8 and CCL4, we start to see more of this steatosis potentially, and we can even outline a little bit of this fibrotic lines. Then we wanted to look at immune cell infiltrate. By doing so, we did an IHC for F480, which is the biomarker for mouse macrophages. And in the no CCL4 groups, we see pretty normal amount of immune cell infiltrate. Same with the no AAV8 and CCL4, we see a pretty common amount. But then when we get to the AAV8 with CCL4 group, we start to see a clear difference in the amount of immune cell infiltrate around these potentially fibrotic bands where we would find. So far, our question is, what is the mechanism of DEC deletion causing exacerbated fibrotic liver injury? So our rationale is DEC is a nuclear protein binding to chromosomes. When DEC is decreased or absent, DNA may be vulnerable to excessive damage. This excessive damage in DNA will result in an increase in hepatic injury and eventually cell death. So to look at this, we first want to assess what the DNA damage is. So by doing so, we looked at gamma H2AX, which is a known biomarker for double-stranded breaks. And so in our figure A in the DEC wild type vehicle group, additionally, we're back to the global knockouts when I'm talking about from here on out. But in this DEC wild type vehicle group, we see no gamma H2AX. In the DEC knockout vehicle group, we start to see a little bit more of this gamma H2AX suggested by the purple nucleus staining, because the gamma H2AX is stained red. Then if we go to the DEC wild type CCL4, we start to see even more gamma H3X present, this is kind of as expected. But again, with the DEC knockout CCL4, we start to see the most amount of gamma H3X, suggesting that it has the most amount of DNA damage. We then did a region of interest assay. Each group had 3 separate samples. And we see that the DEC knockout CCL4 group had the significantly highest amount of gamma H2AX, suggesting that the DNA damage is worse. What's interesting to point out is that the DEC wild type CCL4 and the DEC knockout vehicle group also had pretty comparable levels, which this was pretty interesting to us. Probably warrants more further investigation. So from here, we want to look at if DEC deletion increases cell death-related proteins, which on the Western blots to the left, we first stained for RIP3 and phosphorylated MLKL. These are two proteins required in the necroptosis pathway, a specific type of cell death. And we can see in the DEC knockout treated with CCO4, we see the highest increase in RIP3 and phosphorylated MLKL. Then we want to look at apoptosis markers, which leads us to cleaved caspase 3. And this time we use 4 samples, and we can see that cleaved caspase 3 is a little bit increased in the DEC knockout CCO4 groups. What's interesting too is for the HMGB1, which is a damage-associated molecular pattern, we can see it's a little bit decreased in the liver tissue, which could suggest that it's being released, but this would warrant more further investigation. So this is the full schematic of what we think is going on right now. When DEC is deleted in that first arrow, this could lead to more DNA damage, which could lead to cell death like necroptosis and apoptosis, which leads to DAMP release. This could activate these hepatic stellate cells, leading to more collagen secretion, which leads to worse fibrosis. So summary, DEC deletion, whole body or hepatocyte-specific deletion exacerbates liver injury and fibrosis. DEC deletion increases DNA damage, which leads to an increase in cell death pathways such as apoptosis and necroptosis. I'd like to acknowledge our current lab members. I'd also like to acknowledge Dr. Lisa and Dr. Suzanne for providing our DEC flux and knockout mice. I'd like to also acknowledge the PISA and ASA committee for letting me present today. Thank you.
Awesome talk. We have time for a couple of questions. Do we have any from the audience?
Hi, Ramsey. I can start off. Great presentation. Super interesting. I'm a huge lover of fibroblast work, so I'm all for it. So I was wondering, with the last bit of data that you presented about the cell death and the possible kind of 2 avenues, whether it being a silent apoptotic death or a more inflammatory necroptosis type, do you anticipate it being one or the other that causes a more kind of accentuated kind of fibrosis? And do you have plans to maybe block some of those pathways to see which avenue to kind of go for?
Yeah, it's a great question. So we're still looking at different cell death pathways right now to see if one is higher than the other. What is known with the ALT-AST, there's obviously more liver injury happening because there wouldn't be a release of this ALT-AST. So we know there is more cell death. It could be there's just more cell death in all forms. We haven't found conclusively one is specifically higher than another, but unlike the Western blots shown at the end, they definitely are. We're still looking into if there is one specific type.
I have a question. So you mentioned that DEC is a proto-oncogene, and then you showed that deleting it increases cell death. So I'm just wondering, is this, do you know, or do you have any thoughts on, is this the mechanism by which it is an oncogene? Is it, is it kind of allowing cells to evade cell death when it's expressed?
Yeah, great question. It's kind of it might be kind of context dependent in the oncogenic matter, because it is interesting that we would see more fibrosis when this oncogene is knocked out, which starts off this chronic liver disease pathway, which could end up leading to hepatocellular carcinoma. So it seems like kind of counterintuitive that we would see more fibrosis when an oncogene is knocked out or any sort of damage. So it's kind of too early to make any like decisive decision on what its mechanism is in terms of increasing cancer from this fibrotic aspect we're looking at right now. But yeah, what it seems to be kind of context dependent, I would guess, because it's more studied in its oncogenic effects in other diseases like AML is the big one. But yeah.
Do we have any more questions? If not, then maybe we can move to the next speaker then, who is Anu Balagun, who is coming from the University of Pittsburgh.
Hi, thank you. I'm just gonna share my screen. I think you have to stop sharing your screen, Rona. Thank you. Okay, can you see my screen? Thank you. Hi, everyone. My name is Anu Balagun, and I'm a postdoctoral fellow at the University of Pittsburgh. And this is my thesis work, which I did under my excellent mentor, Dr. Nija Bowen. And the title has changed. It's more mechanistic, so I'll be telling you about inhibiting Wnt-beta-catenin mTOR signaling and how it enhances porphyrin clearance in porphyrias that have hepatic involvement. So this is an overview of what I'll be telling you about today. And if you are a hepatomaniac, then you'll enjoy these topics. And if you're not, I hope that you can learn something new. Okay, so the porphyrias are a group of rare genetic disorders that impair the body's ability to produce heme. And without heme, life as we know it is not possible. Now, heme biosynthesis begins here in the mitochondria with the condensation of succinyl-CoA and glycine to produce the first porphyrin of the pathway. And then this cascade continues until heme is generated. Now, when one of these enzymes is deficient, the pathway gets blocked and the upstream intermediates accumulate, and that buildup causes the clinical symptoms. Now, in this talk, we'll be focusing on the porphyrias that have hepatic manifestations, which usually originate in the liver and which can impact multiple organ systems. Now, we can simplify what happens in the porphyria by imagining the heme biosynthesis pathway as a busy traffic circle, where each porphyrin intermediate is like a car driving along a circular road, and the enzymes that process the intermediates are the traffic lights at the intersection, guiding all of those porphyrins towards heme. Now here, everything's working correctly. All the lights are green, the cars are flowing towards heme. However, when there is a block, the cars which represent the porphyrins begin to pile up, and this is what we call a porphyric traffic jam, which is simply a metaphor for the toxic accumulation of upstream intermediates. Notice PBG here, which is a key biomarker that clinicians use to diagnose porphyria in patients. So this toxic buildup leads to oxidative stress, mitochondrial damage, and disrupted autophagy, to name a few. The porphyrias are not curable. There are supportive therapies like intravenous hemine, but that doesn't address the underlying molecular dysfunction. And there are now recently targeted RNA therapies like givosiran, and it's scientifically elegant, but it's very, very expensive. And in fact, there was a 2020 study that showed that porphyria is associated with a significant financial healthcare burden on the US healthcare system. So we're talking billions of dollars annually. So our goal was to develop novel therapies that have pleiotropic effects on porphyrin accumulation and offer promising options for patients with porphyrias that have hepatic manifestations. Now, we can induce porphyria by supplementing the mouse diet with a xenobiotic compound called DDC, and this causes inhibition and overproduction of heme enzymes, shown here in the red arrows, and formation of this toxic protein aggregate, shown in black or purple. And this produces a toxic hepatic milieu that mimics what we see in patients. Now, we have done a pivotal study in the past that showed that inhibiting hepatocyte beta-catenin could protect mice from porphyria. So we wanted to find out by what mechanism is this happening. Now, I've told you about how inhibiting beta-catenin resulted in less injury after induction of porphyria, but beta-catenin itself is part of the Wnt signaling cascade. It's a master regulator of hepatic zonation, and it controls many other hepatic functions through this Wnt signaling pathway, including through a key enzyme called glutamine synthase, or GS. Now, beta-catenin localizes or is the strongest in pericentral hepatocytes, which are those close to the central vein here. And again, its key target is glutamine synthetase, represented by the GLU gene. And so in these pericentral hepatocytes here, you have expression of Wnt beta-catenin, which leads to expression of GS, glutamine production, and suppression of mTOR, which then in turn suppresses autophagy. So this sort of sets the stage for our mechanistic question, which is, could this pathway be influencing the toxicity we see in porphyria? Now, very, very briefly, autophagy is simply the cell's highly regulated housekeeping system. It just identifies, engulfs, and degrades damaged cellular components. And today I'll be focusing on these proteins, most especially mTOR, which triggers the initiation of autophagy once it's inhibited. So to investigate why these mice seem to be protected from injury after beta-catenin was inhibited, we tested whether blocking Wnt, deleting GS, or a combination of both of these inhibitory mechanisms alters the expression of heme genes or autophagy. Now, if we look at what happens to glutamine synthetase when we delete it or inhibit Wnt signaling, you can see that it's significantly reduced in all our treatment groups, both via IHC, Western blot, qPCR. In fact, in immunohistochemistry for GS, you can see it's classic pericentral zonation, which is lost in all these other groups and is severely diminished after DDC diet. Now, we can confirm that these changes are functionally relevant using a glutamine consumption assay to show that glutamine metabolism is significantly impaired in both GS and the double inhibited livers. And this data basically reinforces the fact that GS expression and function is controlled by Wnt signaling. Now, if we look at mTOR, which, as I said, is the main negative regulator of autophagy, we can measure its activation looking at S2448, and you can see that it's completely abolished when both GS and Wnt signaling are lost. And if we look at it under normal conditions, it's located around the central vein, but also lost in all the other mutant groups despite DDC distorting zonation. So we can conclude that mTOR activation in the liver is heavily dependent on GS and Wnt beta-catenin signaling. Now, we also use spatial transcriptomics, which is simply a technique that allows us to visualize gene expression with spatial resolution across our tissue sections. And to me, it's better than bulk-seq or single cell because you can see the where along with the what. And what we're interested in doing here is mapping those key autophagy regulators, glutamine here to denote the central or the pericentral vein and cells. And you can see that DDC treatment dramatically decreases each of these autophagy initiators that can be rescued after loss of both Wnt signaling and glutamine synthetase signaling. So all of these here are critical for autophagy initiation and lysosomal function. Now we can take a very quick look at the machinery that is responsible for the formation of the autophagosome and the final degradation in autophagy. And here, although these protein levels look diminished, they're actually being rapidly utilized by the autophagosome machinery. And we can confirm this with our spatial transcriptomic data, where you see DDC is severely diminishing these proteins, but after loss of Wnt signaling or GS, we can rescue this. And you can also appreciate here that some of these proteins have pericentral localization. So together we can see that loss of these signaling pathways reactivates the entire autophagic program. Now, there's more evidence that autophagy is actually happening from our TEMs, where we see ultrastructural analysis of these autophagosome membranes indicated by the yellow arrows and yellow boxes. And you can see that with loss of GS or Wnt signaling or both of them, there's an increased amount in these autophagic vesicles just to further bolster our conclusions. So here's what I've told you so far is that in the presence of porphyric injury brought on by DDC, Wnt and GS are usually up, and that sustains mTOR and suppresses autophagy. But if we take away those, we can reactivate the entire autophagy program. So we also looked at the expression of heme biosynthesis genes. And first thing that I really like to point out is that many of these genes are showing pericentral localization, which means they're being regulated by this pathway. And you can see that they're severely increased after DDC, but rescued by loss of that pathway. And we can see functional output by looking at one of those metabolites from the heme biosynthesis pathway via mass spec. And we can see that it's completely lost when any of these pathways are inhibited. So what's happening biochemically is that you have loss of glutamine synthase, which reduces the activation of mTOR and increases hyperammonia. And that loss of mTOR increases autophagy, but that's not enough. So when we take away the Wnt signaling pathway, we can further increase autophagy, reduce porphyrin accumulation, and those overly expressed heme biosynthesis pathway genes. Now, there's evidence that mitochondrial dysfunction in patients with porphyria. So we asked whether this mitochondrial dysfunction also existed with our model and if it's restorable. And yes, if you look down here, you can see DDC does cause mitochondrial dysfunction that can be rescued most especially by loss of glutamine synthetase. And so we also wanted to ask, what about mitophagy, which is basically autophagy, but for the mitochondria. And we used the mitochema mouse model, which expresses a pH-sensitive fluorescent protein, which we can visualize using live confocal microscopy. And if you look here, we can see that DDC severely diminishes mitophagy after these mice have had it. But if we inhibit Wnt signaling, we can actually rescue mitophagy, and you can see that quantifiably here. Now, lastly, we wanted to confirm these findings in humans. So we examined liver biopsies from patients who have porphyria, and we've also seen that, dysregulated or, mislocalized or dysfunctional beta-catenin activity alongside these elevated autophagy markers. And this basically reinforces, that zonated Wnt/GS mTOR autophagy signaling and its clinical relevance. And so this brings me to the conclusion where we can tie the entire story I've told you together, starting out here with this DDC treated injured hepatocytes and turning off the Wnt pathway and its target glutamine synthase to bring down or inhibit mTOR and reactivate autophagy, restore the heme biosynthesis pathway, restore mitophagy and mitochondrial energetics. And then we end up here with a less injured hepatocytes. And with that, I would like to thank everyone that made my PhD possible and made this project possible. And I can take any questions if anyone has.
Thank you, Jemans. That was fantastic work. And again, quite a lot of work. Are there any questions? If not, I have one, and I am very new to the liver. I work mostly in the gut, and seeing the pathway in heme production and that sort of traffic jam idea. Do you know if autophagy plays a normal role in the regulation of that? Maybe removing some of the excess traffic jam. Is that known, and is it are some patients affected by porphyria because of issues like dysregulation of autophagy?
That's a very, very good question. And yes, personally, from reading the literature, I do think so. Porphyria is understudied because it's a very rare disease. So no, we don't have a lot of people. We actually the first group to look at autophagy in porphyria. But from what we've seen, especially looking at the human samples and seeing that they do have dysregulation in LAMP1, which represents the end stage of autophagy, then yes, I would say yes. I would assume that it's just like any other protein aggregation disease, whether it's neurogenetic, in the liver, any other way. So yes, we do think that's what's happening in porphyria, is you have this circle of where they overproduce the toxic porphyrins, and then it accumulates. Then they go to the hospital, they get heme, they get a little relief, they go back, and then it's just a continuous cycle. So yes, I do think so. I hope that answers your question.
Awesome. Thanks again, Anu. Great work. For the sake of time, we will move on to the final talk for this session. So we have Mohammed Nassar from Tulane University.
Thank you so much. Let me share my screen real quick. I hope everyone can see. Okay, so I'm a postdoc at Stony Brook University. Previously, I was in Tulane. The project actually started in Tulane and ended up in Stony Brook. Okay, from Dr. Huang's lab, today we'll be talking about the TED1, which is an epigenetic regulator. Let me give you a background. So ALD is alcoholic liver disease and its burden. So we have like 2 million deaths annually from liver disease. And from these 2 million deaths, 1 million are because of cirrhosis. And this cirrhosis actually, because those 50%, among that 50% is largely attributed to alcohol consumption. And once the fibrosis is developed, it cannot be reversed, everyone knows. What the molecular question is, because it is because of heavy drinkers, so not all of the heavy drinkers have this kind of development. Only 1.6 to 8% of the heavy drinkers develop cirrhosis and these kind of issues. So what is the underlying mechanism? So based on a lot of different mechanisms, we were interested in epigenetics to see if these individuals have different types of epigenetic regulations. So based on the epigenetics, what is it? It's the epigenetic mechanism is basically the DNA methylation, among other acetylation and RNA methylation as well. So 5-MC and 5-HMC, they are the two marks. 5-HMC basically is intermediate active demethylation, and 5-MC is a methylation marker. Clinical findings suggest that multiple studies they demonstrated that human 5-HMC level is decreased in ALD patients. And our study also confirmed these. The 5-HMC were depleted in different ALD liver tissues, which is why we were interested in pursuing the epigenetics side of this liver fibrosis. And what are the TED1 enzymes? TED1 are basically dimethyl dioxygenase, and it performs the catalytic function to remove the 5-MC, which is a methylated group on the DNA, into 5-HMC, which is an intermediate of the demethylation. And basically, we have three family proteins, TED1, TED2, and TED3, and they all expressed in different tissues differently. Our primary study shows that the TED1 over here, when we fed these rats and mice with the ethanol, and when we examined the TED1, TED2, TED3, TED1 being the most consistent among these two groups, and TED1 is the most significant one. Although in our mice model, we also see significant reduction in TED1, TED2, and TED3. Overall, we also see reduction in 5-HMC as well. So we were interested what the role of TED1 is in the fibrosis. So what we did, we have developed a mouse model, which was a completely whole body TED1 knockout. We fed those mice with the alcohol for 8 weeks, and what we found that after 8 weeks, we examined the liver tissue, and we found the collagen deposition in those liver tissues, which suggests the red stain over here suggests the fibrotic scars inside the liver tissues. So we hypothesize, our central hypothesis become that TED1 activity in ALD leads to reduced 5-HMC formation, which disrupts the normal epigenetic regulation of genes involved in this inflammation. And based on our studies, what we did is that we have developed liver-specific knockout model. So our strategy was to develop liver-specific knockout model and check the whole molecular path. And we did the same. We fed these mice with 8 weeks of alcohol diet, and then we sacrificed and we collected tissue and we examined those tissues. First of all, we did the validation, whether or not the TED1 and its activity is reduced. Over here, you can see when we checked the qPCR, we've confirmed that TED1 was reduced in hepatocytes or lower activity. Maybe there are some other like hepatocytes, liver tissue has many other cell types. So we did see some of the TED1 expression to qPCR. These TED1 expression may be coming from the other cell types. But overall, we see 5-HMC was significantly reduced, which suggests that the TED1 activity was really down. And the TED2, TED3 was not affected. And the OUMS29, which are the hepatic cell types, we developed this model and we knocked down this TED1 as well. And we checked the 5-HMC through dot plot as well, and which confirm. And we use this OUMS29 as our in vitro model, and the mice we use as in vivo models. So the first question was, is hepatic TED1 activity suppressed in ALD? To check that, we have collected several human tissue samples and we checked those 5-HMC in those ALD patients, and we see the 5-HMC through dot plot and ELISA was significantly very reduced, which confirmed that hepatic TED1 activity is really suppressed in alcoholic liver disease. And when we compare those isocaloric control with the ethanol-fed mice, we also see that 5-HMC was significantly reduced, and we confirm this with our ELISA data as well. And when we checked the TED1, TED2, TED3, the protein expression was also very down in those ethanol-fed wild-type mice as well, which suggests that TED1 activity really suppressed in ALD case. Now, the question was that does hepatic TED1 deletion exacerbate alcoholic-induced fibrosis? To answer this question, we first, after during the feeding of this, we regularly checked the body weight, and after that, we collected the liver and performed some analysis on that. So the liver weight or body weight in control were the over body weight ratio in control was reduced, and in ethanol fed, this was quite very high, and it was significantly higher. While it was because of the control and the ethanol gives certain opposite results, we were really interested in what's going on in here. The first thing we checked whether or not there is any attribution with cholesterol and serum cholesterol and serum TG levels. So there was no change in serum cholesterol TG levels. We also checked the hepatic cholesterol TG level. We didn't find any changes. The data not shown. And among the ALT, AST, which is the injury markers for liver, so we found that AST was significantly higher and no change in ALT. We further checked fibrosis in those liver tissue, and we found that with staining, we found that the liver-specific knockout model with higher fibrosis, and hydroxyproline also confirmed that, which is a major component of protein collagen if there's a collagen deposition inside the liver. So since the fibrosis is closely linked to chronic inflammation, which drives stellate cells activation, stellate cells are one of the cells inside the liver. Upon that activation, it produces collagen deposition. Therefore, we had to ask, does the hepatic TED1 regulate inflammation and stellate cell activation? To check these two, inflammation and stellate cell activation, first we collected the liver extract, and we found that the two of the key biomarkers for activation of stellate cell, like collagen and ACTA, which is alpha-SMA, was significantly high. And then we checked some inflammation in signs for inflammation. So we checked CD68 and CD45. Both of them were very high in the TED1 whole body knockout and liver-specific knockout. And further histology also showed that there were nucleated cells inside those liver-specific knockout, which suggested immune cells were also there. Upon a certain profibrotic markers like IL-1A, IL-1B, and TNF-alpha was also significantly high in these liver tissues. And now these studies suggest that definitely there is inflammation and cell selectivation going on. So we did some in vitro assays to confirm if there is any transfer of immune cells from the body to this when we knock out TED1. So we did the transfer migration assay, and we knocked down OEMS29 hepatic cell. We culture them at the bottom, and the THP1 cells, which are floating immune cells, and we culture them on the top. And we knocked down TED1 in OEMS. We found that higher transfer of those THP1 cells were into the lower chambers, and we quantified, which was already high. So basically suggests that TED1 inhibition can cause inflammation and cell selectivation, but the real question was, how does it do? Since the activity of TED1 is known as the DNA and RNA methylation, so we have to ask these two how they are doing it. With DNA or RNA methylation, what are the key regulators? Before doing this, we need to identify some target, which target we have to choose, since we have a lot of genes that might be regulated through TED1. So we have to specifically choose which is the suitable target for us to study. So we did a lot of profibrotic gene among 25 to 30. Some of the data is not here, but I'm just showing some of the like the IL-1 beta was significantly high, and in the OUMS29 hepatic cell, when we knock down TED1, we also see that IL-1 beta is significantly high. So we choose IL-1 beta for our further studies. So how TED1 regulate IL-1 beta? There are two mechanisms, either RNA methylation or DNA methylation. Basically, RNA methylation is if there is increased methylation, it would be stability. There would be an issue with the stability. And DNA methylation is about the expression. First, we check the RNA methylation. We use a drug called actinomycin D, which inhibits the RNA polymerase, which because of by binding to DNA, it prevents the RNA polymerase from transcribing the DNA. So the RNA, the total RNA inside the cells, would be subjected to degradation, and we have to monitor by different time period whether or not these mRNA are degraded over time and how fast. So the expected result was whether or not the TED1 knockout has the stability or not. So the data suggests that there is no effect on the stability of these IL-1 beta mRNAs. We further went to check whether or not it's because of DNA methylase. So we use a drug called 5-AZA. This drug basically mimic the function of TED1 by promoting DNA methylation. And when we use the AZA and compare with the DMSO, so since the TED1 knockout increased the IL-1 beta, and when we treat with the AZA, it reduced the IL-1 beta expression significantly, which suggests that it is because of DNA methylation. So can IL-1 beta B inhibition can reverse these TED1 phenotypes in the cells? So to answer these questions, we chose LX2, which are stellate cell and immortalized human hepatic stellate cells. And we first checked, we treated those LX2 cells with TED1 knockout AUMS29 conditional medium. And we found that alpha SMA was high. So that's why we chose this. And secondly, we also used the THP1 cells again, but this time what we did, we performed another transfer migration assay. And in this transfer migration assay, we use IL-1 beta neutralizing antibody and IL-1 receptor antagonist to evaluate whether or not it affects the transfer of the THP1 cells at the bottom of the chamber. And we found that a significant reduction in those transfer of the cells when we treated these two. And that suggests that if we inhibit IL-1 beta, it can inhibit the transfer of immune cells. The next one, the LX2 cells, we also choose this and treat them with those IL-1 beta antagonist and antibody. And what we found that the alpha SMA expression was controlled when we treat with these two agents to control in the LX2 cell. And we confirmed this result with the qPCR as well. And we see no more significant difference in those SMA expression when we treat them with IL-1 beta antibody and receptor antagonist. The next, what we did was to therapeutic rescue of TED1 with the through TED1 cofactor. In this animal model, we use all of the animals are wild type B6. And what we did, we used the alpha ketoglutarate. Alpha Kg is a cofactor for TED1, which we were hypothesized that it might increase the activity of TED1. It will give us certain rescue, and what we did was we used the treated and control group, and the treated group was exposed to and supplement to alpha ketoglutarate, and the control mice and the treated mice both was feed 2% alcohol for 5 weeks and injected CCl4 twice per week. So after finishing this feeding period and injecting period, we analyzed liver weight or body weight ratio was not changed, cholesterol TG was not changed, but ALT AST was significantly reduced with the alpha Kg group, the one which we supplemented with the alpha Kg. And we also checked that IL-1 beta was also reduced in the expression with the ELISA and qPCR. And the 5-HMC was increased and 5-MC was reduced, which suggests that higher TED1 activity. And when we examine with the hydroxyproline and we checked the fibrosis in those liver sections, what we found that the alpha Kg group was significantly reduced. The collagen deposit was very low, and the fibrosis marker was also very low. So in conclusion and in summary, our finding established that hepatic TED1 is act as a critical regulator of ALD progression through its suppression of IL-1 beta expression by modulating the DNA methylation, and TED1 mitigates the alcohol-induced inflammation and fibrosis. It highlights its potential as therapeutic target, and these results not only advance our understanding, but also provide foundation of developing other novel treatment as well. I'm thankful for everyone because it started with Tulane and the faculty of the Tulane. They helped a lot with the projects. Dr. Belong, I think he's listening as well. And my PI, who is moved to the Stony Brook. Thank you, everyone.
Thanks. Great talk, Mohammed. For the sake of time, I think we have the next session has already technically started, so I will hand it off to whoever. But yeah, please direct any questions to the chat for Mohammed.
Great work, everyone. I'm excited to welcome you to session 6 about genetic models revealing mechanistic pathways in ECM organization, hormone signaling, stress responses, organ biology. So I'm excited to be moderating this session alongside Zoe. And first talk for this session is going to be led by Taylor Petrucci from the Medical University of South Carolina. So take it away, Taylor.
Hi, everybody. Let's just give me 1 second to share my screen. Thank you so much for having me today. Let's see. Can you guys see the presenter view?
Yeah, try swap displays.
How's that? Good? Okay. Sorry, I'm in my home setup, so just trying to figure things out. But today I'm going to talk to you about a knock-in mouse model. So very fitting for this session, a knock-in model of dermatosparaxis Ehlers-Danlos syndrome. And let's just get right into it. So dermatosparaxis Ehlers-Danlos syndrome is a very rare subtype of Ehlers-Danlos syndrome. The largest publication and collection of patients only had 20 pediatric patients. It is characterized by fragile skin, severe bruising, and then very distinct craniofacial defects that you can see here on the left of the slide. It's caused by biallelic pathogenic variants in ADAMTS2. And although we have identified the genetic cause, the mechanisms by which these mutations lead to disease phenotype remain unknown. So a little bit of background on ADAMTS2. It's a disintegrin and metalloproteinase with thrombus-bonded motifs too. For the rest of the talk, I will just spare you all and say ADAMTS2. But it's a procollagen 1 N proteinase, so it's involved in cleaving the end propeptide end of collagen so that it can be assembled in, or of fibrillar collagen so that they can be properly assembled into collagen fibrils. It has recently been implicated in other complex disorders, including cancer, cardiovascular, and neurodegenerative disease. This graphic here on the right is from a really interesting review about the functions of ADAMTS2 that just came out earlier this year. And I think it really provides some thought-provoking insights into how ADAMTS2 could be involved in more than just the collagen fibril assembly and may contribute to some of the phenotypes I showed you, especially in craniofacial development and things. So we set out to develop the first genetically accurate model of dermatosparaxis EDS because there are currently no models to study the disease, and I apologize for the graphic images. I'll try and move quickly through this slide. But we introduced a knock-in mutation here, which was present in 3 out of the 15 patients in the largest publication of patients that we've seen. It's introduced an early stop codon upstream of the enzyme active site marked here with the star. And so essentially what you have is a partial enzyme that does not have any enzymatic activity. So it's a completely dead enzyme. And right away, when we generated these mice, as they aged into their juvenile phase, we saw really marked subdermal bruising, really sad spontaneous skin tears, and overall just general features of the disease, shortened snout length. And so this sort of gave us an indication that we did in fact phenotypically recapitulate the disease, but we needed to take a closer look. So here on the right, I have transmission electron microscopy images. This is something that is, or on the right, yeah. This is something that was included in the paper identifying the disease where human patients had these really striking hieroglyphic TEM collagen fibers. So typically you have nice uniform round circular collagen fibers assembled in the skin, and they form a woven pattern. And so you can see in the wild type mouse, you have some longitudinal sections and then some cross sections of collagen fibers. But in our knock-in mice, you lose all of that structure and assembly, and it did almost perfectly match the human TEMs. So then looking histologically at the ECM organization, we have wild type heterozygous and homozygous knock-in mice here. We decided to include heterozygous mice in some of our analyses because there's no data out there suggesting a phenotype in human carriers, but we thought it'd be interesting to take a look. So here, histologically, up top, we have a Masson's trichrome, and you can see that collagen is stained in blue, and you have this loss of sort of the resolution in the tissue staining. So you have it appears to be lighter blue with like a wavy pattern. And then when we stain for collagen 1, this is with the collagen 1 antibody to the telopeptide in collagen, which is immediately adjacent to the part of collagen that ADAMTS2 would cleave. And you can see a nice genotype responsive reduction in collagen 1 expression according with the genotype. So we wanted to take this quantitative data that we collected and get the qualitative data we collected and get a more quantitative picture. The histology told us some information, but we really wanted to be able to make quantitative comparisons. So we used a technology called FibroNest, which is an AI-based digital pathology model. So you upload histological images and analyze how much there is at the tissue level and the general architecture and structure. And then you look at the shape and morphometry of the collagen fibers and overall their relationship to each other. And then you're able to make direct comparisons between your genotype groups and evaluate those using composite scores. So here we can see immediately that the homozygous mice, unsurprisingly, had a reduction in all of the collagen composite stores. So it validated a significant reduction. It validated our histological evaluations and allowed us to quantify the specific characteristics of the tissue that were changed. So fine, assembled, and total collagen were all decreased in homozygous mice compared to wild type. But what's interesting is that some of the characteristics, including especially the collagen architecture, had an effect in heterozygous mice as well. So it again validated this idea that maybe we should be evaluating human carriers for a milder phenotype of this condition. And then taking a look at some of the comparisons that Pharmanis is able to make. In general, we know that collagen is changed, but what about it is changed? So we see this accumulation of fine versus assembled collagen. So the fine collagen here in pink, you have this buildup as we get into the knock-in mice. And this, again, validates what we would expect to see if we have loss of function of the ADAMTS2 enzyme. And then here on the bottom, we have a reduction in collagen fiber density in these tissues as well. But what's interesting is we don't really see that reduction in heterozygous animals, but we do see a start of a buildup of fine collagen. So, and then all of these are quantified on here on the right. So this was really exciting to us. It's a newer technique. It's highly utilized in the digestive and liver disease space, but it isn't something that has been applied very often in skin. And I think it's very informative in terms of going beyond the histology and getting a single fiber resolution to understand what's going on in these tissues. So finally, we decided to do single nuclei RNA sequencing of the skin to show, to look at the transcriptional changes that are happening. What about loss of this enzyme and loss of collagen fiber assembly leads to these systemic manifestations that we see in patients, and which cell types are driving these changes? So when we did our single nuclear RNA sequencing, only one cell population had differential expression of significant differential expression of genes, and that was our fibroblast population. So this confirmed for us that the fibroblasts are likely in the skin are likely driving the phenotype that we're seeing, and that was really exciting because this is the first indication that that fibroblasts are driving this. And so we saw downregulation of important ECM genes, including Col6A2, Col6A3, and fibrillin-1. So we found this very exciting because it's not just fibrillar collagens that are affected. And we were then able to validate these decreases in protein by immunohistochemistry here on the right. So what we've learned from this is that we were able to generate a genetically accurate knock-in model of this disorder, and loss of ADAMTS2 activity did in fact lead to impaired collagen maturation and accumulation of fine collagen fibrils. This was validated using an AI-based digital pathology mechanism, and this gave us single fiber resolution information. Single nuclear RNA-seq revealed that this is a fibroblast-specific driven phenotype, and this leads to downregulation of important ECM structural components. So these data established a mechanistic link between collagen processing defects and fibroblast dysfunction, which provides us a platform for our future studies to test ECM-targeted therapy specifically and try and provide some help to patients. This I just wanted to quickly, before I open up to questions, note that our lab has established a model for DEDS and all of the other 14 subtypes of Ehlers-Danlos syndrome, and these are available for free to researchers. So if you'd like any more information, you can visit our lab's website, and we have transfer agreements to get any research lab set up with these animal lines to help promote research into this rare disease space. So I'd like to thank the members of our lab, our collaborators, philanthropic donors, and then of course, ASIP for hosting me here today.
Nice job, Taylor. For the sake of time, we'll just keep it rolling. So next up, we have Zana Ross from the University of Pittsburgh. Also, Taylor, I guess I should say, it's amazing that you have models of all of the subtypes of EDS. Like, I think that's just awesome. I don't know.
Sorry, everyone, just give me a second.
Thank you.
Oh, there we go, Zana.
Can you guys see my slides?
Yes.
Is it in presenter view, or can you? Okay, I guess I'll get started then. Hi everyone. I first wanted to thank the organizers today of ASIP for having me, and everyone who's here to join in to hear me talk a little bit about my research. So today I will be taking you guys through my work so far studying the gene CREBRF and its role in cardiomyocyte stress and function. So my lab, we're in endocrinology metabolism, focusing mainly on diabetes. And so we had a particular interest in those of Polynesian ancestry because Polynesians are known as the quote-unquote fattest population, maintaining the highest rate of obesity in the world. Our group performed a GWAS in Samoans as well as American Samoans, and we identified a variant in gene CREBRF, which is CREB3 regulatory factor. This variant is a substitution of arginine for glutamine. And this variant is fairly common in these Polynesian populations, but it's virtually non-existent in all other populations that exist, which is just interesting and kind of caught our eye. And so this variant in the GWAS was found to be associated with a very high risk of developing obesity, yet surprisingly, the same individuals with that variant had lower risk of developing comorbidities that are associated with obesity, so lower risk of developing diabetes and hypertension. So looking here at the cycle of metabolic syndrome, which is type 2 diabetes and cardiovascular disease are closely related, we hypothesized that this CREBRF variant could not only reduce the risk of having diabetes, but also protect against cardiovascular disease by improving cardiac health and function. So at the time when we identified this variant, basically nothing was known about CREBRF, especially in mammals. So the first thing we did was we made global knockout models to study like the general systemic effect of CREBRF. To see how CREBRF would affect the cardiovascular health of the mice, we did various wheel and treadmill tests. And in the results, despite having relatively the same feeding cycles, the knockout mice had significantly less activity on the wheel than the control mice did. And in our treadmill endurance test, the knockout mice could not walk for nearly as long as the controls, and they also have dysregulated recovery blood glucose levels. So altogether, these initial studies just show us that the mice with CREBRF expression knocked out develop an exercise intolerance. So like I just said, relatively nothing was known about CREBRF in mammals. but there is a CREBRF homologue identified in Drosophila called Reptor. And Reptor is known to be a downstream effector of the master cellular energetics and stress response complex, mTOR. mTORC1 is important in a variety of heart functions and cellular stress response. So to identify if CREBRF has a similar role of Reptor downstream of mTORC1, we looked at the expression levels of CREBRF under mTOR regulating conditions. So mTOR activities increased by the FED and the insulin addition treatments, whereas mTOR activity is decreased with fasted as well as with rapamycin, which is a direct mTOR inhibitor. So you can see that mTORC1 activity, when increased, decreases the expression level of CREBRF. That is true in our mouse heart models, as well as we have H9C2, so just like cardiomyocyte, cardiomyoblast, neonatal cell lines. And these altogether both show that mTORC1 inversely regulates CREBRF expression. So along with our collaborator, we found that CREBRF expression fluctuates throughout cardiomyocyte differentiation and overall mostly increasing throughout the process. So then we're thinking that CREBRF could maybe play a role in neonatal heart and the maturation into an adult functioning heart. And in our neonatal mouse studies, we examined the cardiomyocytes, and the results are showing here that the rate of death dramatically increases with the knockout of CREBRF, and also that within those deaths, a significant portion are due to apoptosis. So this was super important to us because we see here that CREBRF is required for survival in cardiomyocytes. So neonatal cardiomyocytes rely heavily on glycolysis to meet their energy needs, but then as the cells grow and begin to beat, their energy demand increases. So they switch from glycolysis to oxidative phosphorylation as their primary method of ATP production to meet that need. And PGC1 alpha is also known as a master regulator of this cardiomyocyte maturation, both the metabolic switch as well as mitochondrial biogenesis and sarcomere structure synthesis. And also PGC1 alpha has been implicated downstream of mTORC1 as well. So in our neonatal CREBRF knockout models, you can further see that as expected, pathways related to heart development are downregulated, and those related to heart development and the knockouts are downregulated, whereas those pathways related to heart myopathy is increased. And so this in our knockout CREBRF models had us thinking that PGC1 alpha and CREBRF could both be downstream of mTORC1 and tightly linked to each other. So then we wanted to see if CREBRF is playing a role in cardiomyocyte energy production, and if it does so via regulating PGC1 alpha. So we performed a Seahor assay panel and found that the total ATP production is decreased in these knockout neonatal mouse cardiomyocytes. Glycolysis is increased, and OXPHOS is decreased as well. So we're thinking here that CREBRF plays a major role in metabolic function of cardiomyocytes and maybe even the metabolic switch that occurs from neonatal to adult heart. So in addition, we look to see if CREBRF could potentially be an upstream effector of PGC1 alpha along that mTORC1, CREBRF, PGC1 alpha pathway. So we performed qPCR and Western blot and found that when CREBRF was knocked out, PGC1 alpha expression was down, and also protein was basically non-existent. So we're thinking at this point that CREBRF could regulate PGC1 alpha either directly or indirectly downstream of mTORC1. So at this point in our overall large study, we were thinking that this could be the overall mechanism, just as a visualization for you guys. So cellular stress occurs, affects mTOR activity, which then regulates CREB-RF in one direction or the other. CREB-RF is in the nucleus where it regulates PGC1 alpha expression, and then those downstream effectors in cardiomyocytes for PGC1 alpha occur. But so far, these were all of our knockout models, and we, or me specifically, are mostly interested in the CREB-RF Polynesian variant that I had mentioned in the beginning. So after finishing up those knockout studies, we generated HIPSCs that maintain the homozygous CREB-RF variant, and then we differentiated them into mature cardiomyocytes out to 62 days maturation. And so within our bulk-seq data, you can see that the differentiation that occurs with the progressive decrease of these pluripotency iPSC markers and the increase of these genes that are related to sarcomere structure, as well as ion channel signaling. And then you can also see that CREB-RF and PGC1 alpha both increase in expression throughout the earlier stages of differentiation, and then have a slight taper off towards the end. But you can also see PGC1 alpha seems to have a discrepancy between the variant cells and the control cells towards the end of maturation. So then also at day 62 of maturation, the CREB-RF variant cardiomyocytes are showing less signaling relating to cardiomyocyte illness, I guess, if you will, and an increase in signaling related to cardiomyocyte development and function. And then GO enrichment score also showed that there is increased signaling of pathways related to general cell death, so such as hypoxia, P53, apoptosis, and in this, there was also an increase in glycolysis signaling, which is congruent with the results we had before. So then, based on what we have so far, we're trying to find the mechanism that could explain and encompass the key regulators that we've identified in the CREB-RF pathway and its relation to cardiomyocyte dysfunction and death. So in this process and digging through paper after paper, we identified hyper-O gluconasylation. So PGC1 alpha and mTOR here and here have both been implicated in the cardiomyopathies that are caused by hyper-O gluconasylation, which is post-translational modifications that, when excess, can cause a plethora of cardiomyopathies and also has negative metabolic phenotypes in the heart. So our current idea or proposed mechanism pathway is that mTORC1, whether directly or indirectly, regulates CREB-RF expression, and then under cellular stress, ER stress specifically, the unfolded protein response initiates, where CREB3 is released from the ER, enters the nucleus, where if CREB3 is present, it will chaperone. If CREB-RF is present, it will chaperone CREB3 to be degraded in the lysosome, whereas in the absence of CREB-RF, CREB3, it's been known in some studies to act as a co-activator or co-promoter of PGC1 alpha, but it's also definitely known to interact with host cell factor 1, in which CREB3 recruits host cell factor 1. That complex recruits OGT, and that complex is what ultimately drives the post-translational modification of that O-gluconasylation. So, and then downstream of O-gluconasylation would be PGC1 alpha, which you would end up with the same results regardless, with the increase in mitochondrial biogenesis, sarcomere structure, as well as an increase in fatty acid oxidation and a decrease in glycolysis. So right now we're currently in the process of further studying those HIPSC cells that I had mentioned. We had made with the variant. So we're working on those to confirm or reject that last mechanism with the either CREB3 directly regulating PGC1 alpha or through the hyper-O gluconasylation. And so throughout these experiments, we're going to be looking at various phenotypes such as like survival, calcium flux, metabolic function, things both related to cardiomyocyte-specific function as well as just general cell function. And also in future studies, we actually have human tissue, various tissue samples from Samoan and American Samoan populations. So in the future, once we finish these initial experiments, fully characterizing and phenotyping our HIPSC differentiated cardiomyocyte cell line, we're then going to take our primary samples from the Samoans and we're going to revert those samples to just a pluripotent state and then differentiate and repeat the exact same experiments that we did in our HIPSC cell line. So at the end of it all, I want to thank obviously my lab members who have been so helpful, my mentors who are supporting me the whole way, as well as, again, ASIP and the moderators who have me presenting here today. Thank you.
Great work, Sana. That was an amazing talk, and it sounds like you have a lot of exciting experiments coming up. To keep us on time, if you have any questions for Zana, you can write them in the chat and she should be able to reply. Up next, we have a presentation from Akshata Payadi, who is from the University of Pittsburgh. So take it away.
All right, I'm just going to share my screen really quick. Is everyone able to see my screen?
Yes.
Okay. All right. So good afternoon, everyone. My name is Akshita. I'm an undergraduate from the University of Pittsburgh, and today I'll be talking about my presentation titled Beyond Bile Ducts, in which we investigated kidney injury and other multi-organ effects in FOXA3 Cre-YAP knockout mice. So to start, our lab is generally interested in the study of the liver, more specifically its function, development, and its relation to other systems in the body. And a large function of the liver is to digest fat and excrete toxins. And it does this through the production of bile acids and excreting them through the bile ducts, which is what we study. Now, when bile ducts are underdeveloped, or they just are blocked, then bile acids can accumulate, and this leads to inflammation and fibrosis of the liver, a condition more specifically known as cholestasis. And so this is a model we wanted to study in our lab, and so to mimic those effects, we manipulated the Hippo pathway in the liver. So we essentially deleted yes-associated protein 1, also known as YAP, which is a regulator of organ growth and development. And in doing so, we were able to cause underdeveloped bile ducts in mouse liver, which mimic these effects. And we used FOXA3 Cre as a driver because of its prevalence in hepatocytes. So you'll see here that in the wild type model, all of these branches are a healthy network of bile ducts, but in the mice that didn't have YAP, they have severely underdeveloped bile ducts. And so as we were studying this, we began to see some unexpected consequences of this model in that we were seeing the progression of chronic kidney injury. So in the wild type, the kidney is functioning perfectly fine, whereas in the knockout, we begin to see formation of renal cysts, which begin to severely impair the function of the kidney in the mice. So to study that function more, we tested blood urea nitrogen levels and creatinine levels, and they were significantly higher in the knockout mice, indicating that they had impaired kidney filtration function. And to further characterize this kidney injury, we also stained it with CD45 and Sirius red, which stained for both inflammation and fibrosis. And the knockout models have a much higher prevalence of both of these, indicating again the severity of the kidney injury in the mice without YAP. So this led us to 3 different questions in general. The first one being, what cellular behavior might be contributing to the renal cysts that we're seeing in the kidneys? 2, we also wanted to see where else this FOXA3 is being expressed in the body, since we're seeing unexpected presentation in the kidney. And finally, a more broad question being, does the severe cholestasis impact the kidney and contribute it to the disease, or did we genetically alter the tissue to cause this specific kind of injury? So first, in order to look at what cellular behavior is contributing to the renal cysts, we studied 3 different modes of cellular behavior, specifically proliferation, apoptosis, and primary cilia structure in the kidney. So first, we looked at if excessive cell death could be causing the renal cysts, if cells were dying more right around the area of the cyst, and that was causing these gaps. However, when we looked between the wild type and the YAP knockout using an IHC stain, we actually see no extra staining of the cleaved caspase 3 in the knockout, indicating that there's really no significant difference between the two, which led us to believe that apoptosis wasn't the reason for the cysts that we're seeing. So we moved on to testing whether cell proliferation and excessive cell accumulation could be causing cysts in areas of the kidney. And once again, we compared between wild type and knockout using Ki67 and an IHC stain. But again, we didn't end up seeing any significant difference in cell proliferation between the two either, leading us to rule out this mechanism as well. We then moved on to looking at primary cilia structure abnormalities in the kidney. So primary cilia in the kidney help with kidney signaling. And oftentimes with renal disease, if there is an issue with the structure, that can affect signaling, causing more kidney injury. So here, the primary cilia are marked in white. We used immunofluorescence staining. But again, there was no significant difference between the wild type and the knockout, leading us to believe that these primary cilia structure was not the cause of these renal cysts. While we can't conclude anything about signaling, because signaling could still be altered regardless of if the structure is fine, we can conclude that structure itself is not causing this issue. So while we weren't able to find a specific reason of cell behavior that causes this, we were able to rule out several viable reasons as to why these cysts are being caused. So from here, we moved on to try to learn where else the FOXA3 may be expressed in the body. And so to answer this question, we gave attention to systems not only developed by the endoderm, like the liver, but also the mesoderm. So when we did this, we used TD tomato reporting mice to highlight where the FOXA3 promoter was being expressed. And in both pictures A and B, we first see it in the kidney, the renal cortex, and the renal medulla, like we were expecting, since that is where we saw the primary kidney injury. But to our surprise, we also did see this in the reproductive system. Specifically here, we have the male reproductive system of the mice in the bladder and the testes, which we were not expecting because these are developed more from the mesoderm. And so this is highlighting multi-organ consequences in other systems. And this is further backed up by the fact that our YAP knockout mice actually lacked Leydig cells, the male mice, and they weren't able to produce mature sperm, meaning that they were sterile, which is an effect we were not expecting to see. And so this gave us evidence that we did genetically alter the tissue in a way we did not intend to by taking out YAP in the way that we did and at the time period that we did. And so this leads to altering our final question. Instead of questioning whether we did alter the tissue genetically, we now know that we did. And so we come back to the question of whether does the severe cholestasis impact the kidney and contribute to the disease, or is it our genetic alteration that's doing so? So to thoroughly answer this question, there is more work that is going to need to be done, but there are several ways that we can study this. And one, it would be to observe kidneys in other cholestatic models. If we see this kidney injury in other models, then this can help us determine that it is due to the cholestasis and not what we did genetically. Something else would be to promote bile acid excretion, which would mean unblocking and allowing the bile acid to be excreted, which would minimize the effects of cholestasis. And if we still saw kidney injury through this, we could determine that the kidney injury is caused because of something that we did genetically. So originally, while we were intending to study the importance of bile duct function in the liver, this emergence of kidney injury really underscored the broader development of YAP in the body. And we were able to discover that the FOXA3 Cre-YAP knockout mouse model really highlights this multi-organ consequence of YAP loss with disruptions in hepatic, renal, and reproductive development. So from here, there's a few different avenues we would like to go. The first really highlighting what is causing this kidney injury and whether it's due to this model of cholestasis or this FOXA3 mediated YAP loss. Something else that we would like to do is to study other behaviors of cell pathology, which are directly linked to renal cyst formation, because if we do that, then maybe we can mitigate the kidney injury separately. And finally, we would like to explore other interventions that may mitigate this renal damage as a whole, whether that is through drugs or any other means that we are able to find. Okay. And I just, to end, I would just like to thank everyone in my lab, my PI, my mentor, and ASIP for hosting this today. Thank you.
Nice job. I'm going to start with a question. You had mentioned that the original purpose of your project was to look at the bile duct. So, and it was almost unintentional that you found the kidney injury. So I'm wondering if there's a well-established association between the loss of the YAP protein, even in other tissues. Is that something that's well established, or is this also relatively new?
Yeah, so based on what we were looking at, this seemed relatively new, which is why we were very surprised at what we saw. We weren't expecting anything in the kidney or the reproductive system because they're more derived from the mesoderm, and the liver, and these things are more from the foregut endoderm, which is what we wanted to study specifically. And so what we were thinking was maybe it's the time in which we deleted the YAP, which was in very early development, that could be causing overlap between the endoderm and mesoderm derived structures.
Right. Okay. Awesome.
Cool.
Are there any other questions?
Yeah, we have another question in the chat from Vic Meadows. She said, beautiful presentation. Is there a way to have ectopic YAP activation in utero or in young mice?
Sorry, could you repeat the question? I was just trying to find it myself.
She said, is there a way to have ectopic YAP activation in utero or in young mice?
Honestly, that's not something we've done yet. And I don't think we've looked at the literature like too much in that regard. But I think that's definitely something we should try to do. But the mice that we have used were pretty young, so I do think it's something that could be done.
Great. I think you have another question from Cole, but for the sake of time, I think I'll let Zoe introduce our last speaker for session 6. Great job.
All right. Yes, last but not least, we have Sydney Severance from the University of South Carolina, Charleston.
Hi, here, let me pull up my screen. I'm Sydney. I'm a junior at Vanderbilt, but I'll be presenting on my research at the Medical University of South Carolina in the Norris Lab from this summer. So this summer, I worked in the Norris Lab, and I worked on a project related to hypermobile Ehlers-Danlos syndrome, or HEDS. And HEDS is a heritable connective tissue disorder that affects multiple organ systems. and presents with a wide range of symptoms, including joint hypermobility, pain, autonomic dysfunction, and it's frequently associated with the immune-related conditions, including allergy-like symptoms and mast cell activation. As you can see, about 40% of HEDS patients have a diagnosis of mast cell activation syndrome, and almost 90% have at least one allergy-related disorder, but the relationship between HEDS and like mast cell issues or allergy and issues is really poorly understood. Also, it's important to note that 97.7% of HEDS patients are female at birth, and symptoms typically flare around, puberty or hormonal changes. So this overlap with immune allergy issues and then hormones led us to investigate how sex hormones may influence mast cell activation as a potential contributor of the symptoms seen in HEDS. This slide really just serves to show like all the different things that HEDS can impact. And, for today, I'll just focus on the mast cell activation, but this condition can really have tons of different impacts beyond that. Okay, so this is a video my mentor took, and I thought it was really cool because it just shows the mast cells actually degranulating. And mast cells are a type of immune system, and when they're activated, they release mediators like histamine, tryptase, chymase, and other enzymes. So I thought that video was really cool when I was learning more about it because you could really see it actually degranulating. So our first goal was to characterize the hormone receptors present in Levy cells, which is the type of mast cell that we used. It's an immortalized mast cell line. And we just wanted to start by characterizing like what hormone receptors were expressed. And specifically And so we wanted to see if they were like nuclear or membrane receptors. And so first we isolated RNA to do RT-PCR. This is kind of an overview. It was more for my sake when I was learning about this, but genomic signaling, of course, is when the hormone binds to a nuclear receptor and acts as a transcriptional factor and then regulates the gene expression. And then non-genomic signaling is when the hormone binds to the receptor on the membrane and activates like the secondary messengers. And this is a much faster response. So we wanted to figure out if it was impacting this, like which pathway, since it's just really not understood in Levy cells. So for our positive control, we used normal human dermal fibroblasts just to make sure that all of the primers were working right. And we did RT-PCR for all of these different hormone receptors, and the ones with stars are the nuclear receptors, and the ones without it are the membrane receptors. So we found that estrogen receptor beta was present in the Levy cells, and that's a nuclear receptor. And then PGMC1, PACR5, and PACR6, which are all membrane receptors, were also present for progesterone. And then we wanted to double check that beta estradiol receptors were present in Levy cells, and we did that with Western blot right here. We also found that androgen receptors were not present in the Levy cells. Then we did immunocytochemistry, and this really double confirmed the absence of androgen receptors in the Levy cells right here. We stained for tryptase, which is released by mast cells, and then androgen receptor. And as you can see, there is no androgen receptor present, which we expected, but it was good to confirm that. One interesting thing that we found was that when we stained for aromatase, we saw aromatase in the Levy cells. And this is really interesting because aromatase converts androgens to estrogens, which means that even in the absence of androgen receptors, androgens could still be influencing mast cell behaviors indirectly by being converted into estrogens. But more research is definitely needed on this before making any definitive claims about aromatase in Levy cells. Then we moved on to do beta-hexosaminidase assays. And beta-hex is an enzyme that's released during the mast cell degranulation, so it could be used to quantify how much it's degranulating. And the main thing we wanted to test was what would happen when the mast cells were treated with hormones in a 90 90 minute incubation period and then treated with a trigger. Like, would they be stabilized by some hormones? Would they be activated by others? Or would there be no impact at all? And so there were a lot of steps to set this up, and we had to figure out, the correct doses of the hormones and the trigger. For the trigger, we used compound 4880. So I'll show you some different graphs about how we decided on what doses. We first wanted to pick a dose for each hormone that wasn't going to kill our cells and skew our data. So first we did that, and this kind of just shows the hormone viability in the first 30 minutes after they were treated with the hormones at different doses. So this helped us really like narrow down what dose we were going to use for the beta hex assays. For example, with progesterone, we ended up using this dose right here. And that was the hormone that I mainly focused on this summer. But next, we wanted to make sure that the hormones themselves didn't cause the mast cells to degranulate, because that would really confuse our data a lot. And so we ran a beta hex to see if the hormones made them degranulate when it was just the hormones and the mast cells. And there was no degranulation that was significant happening, which we expected, but it was good to, it was really good to confirm that. And then the last step before running the beta hex that we had set out to run from the start was to figure out what dose of compound 4880 to use. And so compound 4880 is a chemical agent, and it's used to trigger the degranulation of mast cells. And it works by activating G proteins on the mast cell surface, which leads to a release of histamine and other inflammatory mediators from the granules inside the cell. And as you can see here, the doses of both 10 and 100 cause significant degranulation. So we decided to use those 2 doses. Okay, and then we finally actually got to do the beta hex and measured the percent degranulation of the LUVA cells after they were treated with 10 micromolar of progesterone and the 2 different doses that we landed on for the compound 4880. And we saw that at lower doses of 4880, the progesterone had a stabilizing effect on the degranulation, and it seemed to degranulate less, but it wasn't statistically significant. It was close, but not quite. And there was a trend towards significance at least. And so we found this interesting because we were like, maybe if we left the hormone on for longer or set it up slightly differently, or maybe used a different type of media with the mast cells, we would have seen slightly different results. So this was super interesting to me, and I would have loved to research it further. Then we did cytokine arrays. Cytokine profiles are impacted by the different treatments with the different hormones, but we found that very few cytokines actually reached a statistical significance. We also thought that maybe the timeline of the treatment and the assay sensitivity could have been a limitation here. We didn't really find anything super telling, especially on progesterone, which was my focus. We did find that 71% of the cytokines for progesterone were downregulated, suggesting, maybe there was some sort of anti-inflammatory connection there, but most weren't significant. Okay. Finally, we did cytoplasmic and nuclear extractions, which I personally really enjoyed learning how to do. And the LUVA cells were treated with the hormones for 30 minutes. In this case, we did beta estradiol. And then we did the cytoplasmic and nuclear protein extraction. And the goal was to make sure that we were using the right dose of hormone treatment before sending for bulk sequencing to look at gene expression. We just wanted to make sure that it was actually enough of a dose to be making a difference. And we saw a decrease in cytoplasmic beta estradiol at increasing doses of the estrogen treatment, which confirmed that it was, entering the cell, engaging nuclear receptors, and it was an effective dose for downstream gene analysis. So in conclusion, we found that the membrane-bound non-genomic progesterone receptors detected in LUVA cells, that was the pathway. And so progesterone reduced mast cell degranulation in LUVA cells, supporting, that mast cells could be stabilized by progesterone. But also we couldn't really quite say that because it was just trending, not technically significant. Also, these findings could help explain hormone-sensitive flares in different conditions like HEDS, and it could have broader relevance to immunology, women's health, and hormone-linked autoimmune or inflammatory diseases. And so for future directions, we want to try different doses, different time courses, like I mentioned, and identify the exact receptor mediating the progesterone's effect. And then, of course, characterizing, downstream signaling pathways and testing other degranulation triggers was something that I was super interested in too, because we did the compound 4880, but is it specific to just that trigger, or would these results be seen in other triggers as well? And then we would like to validate these findings in primary human mast cells, and then eventually, like potentially HEDS, do more with that in mast cells in people with HEDS. So I'd just like to thank the Norris Lab, where I did all this research this summer, and my mentors. It was great. Thank you.
Awesome work, Sydney. We have about 4 minutes for questions if anyone has any questions for Sydney.
Hi.
Oh, perfect. Good friend.
Yeah, I have a question. Great talk, Sydney. So my question is, like, when you treat the cells with the hormones, do you pre-treat the cells before adding the compound, or you add both at the same time?
We added the hormone, and in some cases we let it sit for 30 minutes. In other cases, we did 90 minutes, and then we added the degranulation trigger for typically 15 minutes, and then would run the beta hex assay or do the next part of the experiment.
Great. Thank you very much.
Sydney, did you ever, I know you're still like optimizing things. What do you think about adding multiple hormones at once? And I know that women are more affected, but have you had any interest of maybe adding like testosterone or something like that? And what do you think you would see?
I think it would be really interesting. Of course, the internship I did in the lab was only 10 weeks, so I did not have enough time to explore all of these things. But I think it would be super interesting, and maybe it would also stabilize the degranulation. But I guess I don't have the knowledge to say for sure what it would do, but it would certainly be super interesting to try that.
Awesome. Does anyone else have any questions for Sydney? If not, you can write them in the chat. I would like to congratulate everyone. That was a great session. I learned a lot, and thank you, Zoe, for helping me moderate. I think we're gonna have about a 10-minute break, but don't go away because we have a very special session on trial tribulations and triumph. So we have two invited speakers, Dr. Evan Delgado and Dr. Brian Wakefield, be speaking about their experience. And so don't go away, but we're going to have a nice little break. Thanks, everyone. Lisa, we can see your email now.
Somebody needs to answer a couple of them. Thank you.
Of course.
Okay, hi everyone. It's 3 p.m. I hope you're all back and you had a quick, nice break. We are going into one of our special sessions titled Trials, Tribulation, and Triumphs, Non-Traditional Paths to Scientific Success. It will be moderated by myself and my wonderful colleague, Anna Tengler. And we are very excited for this session because we are always talking about the successes in science, and some people have a much more complex path to success. And I think that in the situation that we are now, many of us are experiencing that. So we've picked some absolutely fantastic people who are exceptionally successful but have had non-traditional paths, and we hope that you can learn a lot from them as much as we have. I think the whole of ACP in general, the faculty are always very willing to be good mentors, and so are these two people. And so without too much talk, it's my very, very great honor and pleasure to introduce Dr. Elvan Delgado, who used to be from the University of Pittsburgh, but is now from the Medical University of South Carolina. So please take it away, Evan. And also please hold all questions till both speakers have completed their talks, and then like we will have an open session. Thank you.
Hey everyone, and thank you, Anu, for including me in this session. Yeah, like you said, I feel like my path has been traditional, non-traditional as you can go. But my experience is an N of one. So hopefully I can give you all a little bit of a fresh perspective of like what happened to me in the past 20 years or so of my career. And with that, I just have a little, like one little slide that I was hoping to share with everybody. Um, let's see. This hopefully will give you an idea. Okay. Of my career path over the past 20 years. And you all can see that, right? Okay, great. So it all began in 2005 when the most popular cell phone was the chocolate razor. So that's when I got into my undergraduate school at the University of Albany in New York, and that's where I did my bachelor's. And I know that we have a whole mix of undergrads and grad students and postdocs in the audience, so I was giving you a little bit of background of where I came from. So there I did my major in molecular bio and biochemistry. It was more of like a chemistry-based degree program. So when I started entertaining the idea of going into a PhD program, I felt like I was probably a little out of my depth. Uh, but luckily for me, I was able to successfully gain entrance into the PhD program at Pittsburgh in 2009. And I learned really quickly that I needed to step up my game in biology. I was really good at complex mechanisms and like biochemistry and things like that. But when it came to the knowledge of things actually signaling, like proteins actually talk to each other and they do things, that was not built into my undergraduate education, which was, really interesting. So at that point, I rotated in a bunch of labs for about a year, and then a year later, I joined the lab of Dr. Paul Munga, who many of you probably know pretty well. And there, I did my PhD with Paul. I became a member of ASIP, I think in 2010, and I've been here ever since, basically. So once at that point, I published a couple papers. I wrote an F. Didn't get funded, but then eventually I defended my PhD in 2014. And then at that point, I was like, okay, so what am I going to do next? So when I got in, uh, when I was provided entrance into the actual defense part of my program, I started entertaining different postdoc positions because I wanted to do academic science. That's what I knew what to do. And I really liked it. I really liked the work style. I really liked being able to ask my own questions and, answer them and work on what I thought was important and help out other students and other trainees and just continue to work in academia. So that was kind of like where I fell into my interests and my passion. So I was entertaining a couple of different postdocs, and actually at the University of Pittsburgh there was a position available in our drug discovery institute. So my background was in liver cancer and liver biology when I was working with Paul. And I thought like, well, what better way to kind of like enhance my, experience by like working on drug development and drug discovery. So I matriculated into a postdoc position with it was a joint position with the McGee Women's Research Institute and the Drug Discovery Institute. So I had two postdoc PIs basically. So Steffi Osterich was the faculty member at McGee, and Mark Scherdeck was the faculty and the Drug Discovery Institute. And there I was working on a project with ovarian cancer. And I quickly learned that it was really difficult to manage a situation where the two PIs didn't really quite communicate very well with each other, and I was the go-between. And I thought that it probably wasn't the best environment for myself. And I tended to get put between two different situations where on one side I was being treated like an academic, like here's like 5 different directions, work on all of them. And oh, if one direction like really pans out, let's focus on that, like full bore. And on the other side, we were working on a project that we can only work on this, we're only funded for this, make this work no matter what. So it was like it was hard to balance that between the two different positions being one position. So with that, I published the work that I was working on in the year that I was there, and then I found another postdoc position with another member of ASIP that some people may be familiar with, Andy Duncan, who had an opening in his lab. So in 2015, I joined his lab, and I helped finish up a paper that they were working on. And then in the next year or so, after that paper was finished, I applied for an F32 award. And this is where I, really focused on like liver biology because I, again, had a background in cancer. So I thought that, well, I really wanted to work on more like normal biology, seeing how the mechanisms that might go wrong during normal biology can, lead to cancer and carcinogenesis. So that's kind of the idea behind the F32 application that I put in here. That wasn't discussed. So then I was already my second year of postdoc, basically entering into the third year. So I was like, okay, let's just circumvent that and go for a K application. So in 2016, I applied for a K application at the end, and then again, another K application in 2018, another K application in 2019, and you can see that it was iterative. I kept applying, I kept applying, I kept applying, but nowhere in this stage I really found a lot of success. They kept getting better. I kept getting really much better comments. I kept getting better scores, but nothing was ever pushing me to the fundable rate. Um, and then in 2019, I submitted my final KO1 application because here I was getting kind of towards the end of my, eligibility for being a K applicant. But I was applying a cancer project, a liver cancer project, to the National Institute of Digestive Disease and Kidney Disease. And at the time, the program officer that was in charge of that mechanism reached out to me and he told me, "Oh, you should really withdraw your application. We do not like to see anything to do with cancer." And I was like, "What? All the previous applications that I had put in were all related to cancer in some way, shape, or form." And he was like, "Oh, well, if I would have seen that, I would have advised you the same." And I was like, "I'm pretty sure you were my program officer before." But so what I did was I tried to get the K application transferred over to the NCI, but they didn't have the same mechanism. So I withdrew that application. And then when I talked to a program officer at the NCI, they advised me to apply for a K-22 application, which allows you to apply up to 8 years of postdoctoral experience. So, and the K-22 application it's not like a traditional K-01 or a K-99 that everybody always talks about. It's more like a career transition award. It's for those senior postdocs that are trained enough. They have the training, they have the faculty backing, and they have the know-how, they have the publication record, but they just need a little bit of independent bump to get them to a faculty position. So what the NCI would do is they would release the funding from the K-22 if you have a tenure track faculty position. So I applied for the K-22 application during the pandemic, and then I published a paper. And because I was coming up on my eligibility for all these K awards, I thought, okay, I can't really be a postdoc anymore. I feel like I need to make the next step in terms of like how can I keep moving forward? How can I keep, you know, making myself eligible for grant funding? So, and that's when Paul had an opening in his lab for a research assistant professor position. So there he brought me in 2021 after I had resubmitted my K-22 application. And then 3 months after I joined his lab, I got a fundable score on my K-22 application. So I was really excited about that. This was the first time I ever had any kind of experience getting anything funded, and I was excited. I didn't know what to do next. Um, and so the next thing I did was I waited. I waited. I contacted the NCI. Like, what do I do? Who do I talk to? What do I do? And like a year went by before I had any other notice. And then almost another half of a year went by. So now we're talking close to 2023. And then a new program officer reached out to me, and a director of the grants program reached out specifically to me, and they apologized because my program officer that was in charge of my program left the NCI, and his portfolio, which included my application, went completely unnoticed. So it was at that point where they apologized and they wanted to disseminate my funding as fast as possible, which again, as if you remember me mentioning, required me to have a tenure track faculty position for them to disseminate my funding. So I didn't really have the true opportunity to do the faculty search when you solidify a K award. So I talked to Paul, I talked to George, who was the department chair at the time, George Michaelopoulos, and I was able to kind of go through the rigmarole of being appointed a tenure track faculty position in the Department of Pathology in Pittsburgh, which allowed me to attain my K-22 funding in 2023. So you can see that was like almost 2 full years after I got a noticeable or a fundable award score. So it was at that time where, I started working on my K award. I had a kid, and I kept working, and I was at this point where I just felt like, as you can see, my entire training, my entire career history has been at Pittsburgh. And I felt like all the rumblings of people saying like, wow, you've been here really long, or like, you've really been at Pittsburgh your like whole time. That, it was starting to not sink in, but make me think like maybe I should try to think about, what to do next after Pittsburgh. Maybe I need to put myself in a different position where, I can learn from other people and other people's experiences. And that's when I was actually recruited this past year to MUSC, where I started my position as a tenure track faculty position in the Department of Regenerative Medicine this past September. So now I have my growing lab. I just hired a lab manager. I'm hiring a junior lab tech. I'm getting 2 rotation students potentially rotating with me for the next couple of months. And I've got great company around me, as you've probably seen by yesterday's and today's seminar or presentations by being presented by a couple of, the faculty members that are here at MUSC. So I'm really excited to be in this new environment and trying to kind of push my own career, my own independence forward. And with that, I guess I could probably leave some time for questions. That's basically my experience over the past 20 years. There's a couple of little like, personal anecdotes that went into all these decisions, obviously, but majorly like that's how my career kind of progressed over the past 20 years or so. So yeah, thanks for letting me verbal diarrhea at you about like what I've done in science and my career, and hopefully giving you a little bit of inspiration about, sticking things out. You could see that when I was with Andy, I postdoc for about 6 years or so, and that's people don't like to hear that, but it happens. You in your head, you don't think that you're going to postdoc for that long, but I was productive in that time. I published, and I got intramural grants, I got things. I was successful. I felt accomplished, but getting those major things, was kind of like the thing that I really kept wanting to shoot for. So yeah, thanks again for, inviting me and letting me talk to you all about my experience, and I'll be happy to take any questions in the end.
Thank you very much, Evan. That was absolutely fantastic. I'm sure there'll be lots of questions. I'm going to hand it over to Anna for the next wonderful, wonderful talk.
Thank you, Dr. Delgado. Like Anu said, that was a great talk, but I want to hand it over to our next speaker, Dr. Brian Wakefield from the University of Wisconsin-Madison.
I think you're muted, unfortunately.
I did the old person thing. I'm sorry. I'm Dr. Brian Wakefield. I'm here at the University of Wisconsin-Madison's chemistry department, and I'm going to talk about a series of unfortunate events, my time in academia. So kind of laid out all the unfortunate events. Hopefully I get this to work. Okay, I'm being very old. I'm sorry. Let's try again. I'll figure it out, I promise. Maybe I will. There we go. All right, so the unfortunate events kind of run all the way from undergrad, all the way through my current position. I'm going to focus on the things that kind of went wrong and the things that I got out of each position. So undergrad, this is where I kind of decided to pursue chemistry. My path there was I got bad grades in the other sciences, and I ran out of other sciences. So you may have heard D is for degree. For me, D was for done with that major. I got a D in biology, which is very disheartening to this group, and a D in solid state physics. So I was just left with chemistry, which I think all in all ended up being a good choice. The only kind of bad thing that happened in undergrad was I was booed at my own graduation when they announced that I was going to the University of Pittsburgh. So if you, as Dr. Delgado knows, West Virginia University and University of Pittsburgh don't get along well. So then I went to the University of Pittsburgh. Like many of you, it was long hours and too many years. I often ask what happened to my 20s from graduate school. My kind of publication track record was I had my first project, I ran it all the way through to the very end, the last step, and that last step did not work, and that work was never published. And then I published a synthesis, and in my last 6 months to a year, I improved that synthesis. I probably cut it down to like half its length, but it also was not published. So I had a student many years later go to the University of Pittsburgh for graduate school, and they talked to someone from my former group who said, oh yeah, I've seen his thesis. It's full of unpublished results, which is not really what you want for your thesis to be known for. My lasting contribution to the group was this picture of my messy hood from a Saturday afternoon. It is still on the departmental the group website as the what not to do 17 years later. I just pulled this picture this week. On a personal note, I defended my thesis with cancer. This is a picture of me post-op when I was correcting my thesis on pain meds. And I graduated in 2008, which means there was no economy. I was thankfully able to get a postdoctoral position at Emory University. So I moved to Atlanta, and I got there in August. And I found out in November that the grant that was funding me did not get funded. So there was no funding for my postdoctoral position. So I started kind of applying for jobs. My PhD advisor was like, why are you applying for jobs? Basically, what did you do wrong? And I was like, I didn't do anything wrong. I was able to find another lab that paid for me for a year to do some work for them, but my postdoc position ended up only being about 15 months before I needed to find something else to do. The good things that came out of being at Emory was stressful due to like the lack of funding, but I really got to grow my love of teaching. I ended up being kind of the unofficial tutor for the incoming graduate class. So I would go over to their houses, they'd feed me dinner, and I would be at a whiteboard kind of walking through what they were doing in their introductory like graduate courses to try to help them out. On a personal note, I did meet my future wife, so that was good. And just as an unlucky thing, my car got crushed by a tree one day when I was at work. So, you know, things happen. Then when my postdoc ended, I kind of scrambled and got an adjunct teaching position at Georgia Perimeter College, Newton. This branch of these colleges don't exist anymore. They've been like bought up by the Georgia state system. But this is where I kind of first got to teach my own class. So I taught Gen Chem 1 and then 2 sections of nursing chemistry. My teaching schedule was I taught from 11 a.m. to 9 p.m. on Monday. And then 11:15 to 12:15 on Wednesday, which doesn't sound that bad until you realize it was a 45-minute drive to campus each way. And I made $5,000 teaching 3 classes for that semester. Thankfully, my pipes only froze once because I wasn't able to heat my house. And my then girlfriend, now wife, would take me to Moe's for a burrito. That was my treat for like surviving the week. And this is where like I have one very distinct memory of like working with a student who really wasn't sure if they were like ready to go on to like the University of Georgia after their community college time. And I like feel like I really helped them and like built their confidence. So it's like something I will take away from this experience, like working with that student. So from there, I started my time on the tenure track at Delaware State University, which is a small HBCU in Dover, Delaware. I learned how to teach. Like I was kind of just thrown in and had to teach my classes, and I had learned how to adapt my teaching style. The students were good. In the course I was teaching organic chemistry, they weren't used to a lot of rigor in the previous years. And so after my first exam, there was a bit of a mini revolt. There were 8 to 10 students who went to my department chair and demanded my firing. I didn't get fired. And I think by the end of my time at that university, like that group of students and then my next class, I think I reached them pretty well and was able to make an impact on them. I also became the director of the graduate program for a year, and I had to deal with multiple plagiarism issues, which led my office to be vandalized. And then I was threatened by a student's brother after a meeting about them about the plagiarism that was going on in their thesis. I was way too young to be the director of the graduate program. I was like 31. I'd been there for a year. I really didn't have the like experience I needed to kind of like handle that kind of position, but I learned some things from it. And then I ended up leaving. Oh, I should say, I did get to start my own research lab. So this is where I got to start mentoring undergraduate research students. So this is my first group. This picture was taken with a potato back in like 2010. And I'm still connected with some of these students to this day. I have a student who's currently in medical school that has checked in with me every so often. And then I left due to kind of the lack of an NMR, which is kind of an important instrument for an organic chemist. That'll come up later in a second. So then I went to Coastal Carolina University. So there from 2012 to 2024, I earned full professor in 2023. I had really excellent colleagues. That's something I can really take away from that experience. I have a lot of good friends there. Like one of my largest accomplishments was I helped start the CCU IMBRI program, which is an NIH-funded program that supports summer research for students in the biomedical sciences. And we trained over 100 summer students in my time there. The program's still going. One of the last things I did was get it renewed before I left. I secured a bunch of instruments for the university, got us a new NMR, tissue processor and cryostat for my biology colleagues, helped get a microscopy system on campus and a solvent system for some of the synthetic people there. And then I helped restart the chemistry club and kind of give the students in our department kind of a feeling of community and belonging. And I helped mentor over 50 students and decide their futures, one of which is our esteemed moderator, Anna, worked with me. And then some of the challenges I faced, we'll go back to the NMR. I had to conduct research with an NMR for 6 years. An NMR is kind of a daily use instrument for an organic chemist. So I would make a 4-hour round trip to Charleston to use an instrument at the College of Charleston about once a week to be able to get data to know what we were doing. There was kind of limited funding and support for research, especially when I started. So basic things you take for granted, like ordering and grants, were really challenging at times. I had a stalker at one point. 0 out of 10 would not recommend. That was not a fun semester and a half to year. That was no good. And then I came to the University of Wisconsin-Madison last fall. I'm the director of undergraduate chemistry research. I'm the only person with that kind of job title on campus. So I oftentimes find myself dealing with students from other departments, which is cool. I think the really good things about this is I get to support a larger group of students getting into undergraduate research and kind of navigating that. This was our poster session for undergraduate research last spring. We had almost 50 posters of all chemistry students basically presenting their research. I get to run an undergraduate research space in a very new building. They had built this lab space, but they didn't actually have anyone using it. So since I've been here, I've been able to work with students to give them an opportunity to build their resume before they go out to try to get faculty, to get lab space in faculty labs, or to kind of catch students that didn't get a research opportunity before they graduate. So getting seniors so they have a chance to like put on their resume that they did some research. And I'm like learning I'm like trying to improve some departmental policies around research. That's kind of my gig now. Kind of the challenges is I spent a very long time being a faculty member, and I'm now more in an administrative role. I'm just trying to learn how to do that. And every university you go to operates differently. And so kind of navigating that administrative labyrinth is difficult. And then on a personal level, I'm kind of dealing with not teaching on a daily basis. Like a lot of my identity was tied up in being a faculty member and teaching, and kind of learning how to be more of an administrator is something I'm working through now. So kind of all this stuff, was it all worth it? I think yes. My career is going to really be defined by the students I've worked with. So I worked with 76 students who've gone on to get PhD, PharmDs, MDs, get jobs. So this is just kind of a sampling of some of the students I worked with. Again, we got Anna down here in the corner. And then I taught organic chemistry to hundreds of students, and I hope I was able to like make the subject matter a little bit more appealing and a little easier to understand for them. And a lot of the challenge I faced kind of dealt with research and like research infrastructure. So I wish, like, to this point in my career, I'd been more productive as a scientist. Looking back on it, I think there's more I could have done. And I do think that some of the situations weren't great to kind of support me. But I haven't retired yet, so I still got time. And then I think for all of you, like find your joy in whatever position you're in. So everything that you do in your career is going to have challenges. Every position, as a graduate student, an undergrad, early faculty members, right? Find the aspect of your position that you find rewarding. It's going to make you feel successful and fulfilled, right? Like there's going to be stuff you don't like, but like really focus on those things that you do like. And nowhere is perfect. Everywhere that you work is going to have issues. And so kind of determine what issues you can deal with in any given situation. If you see problems or issues, determine if like you can work with them, or if you need to try to go out of your way to fix that problem. And if you feel like you need to fix it, you need to step back and think, is this something I am able to fix? Because you can put a lot of effort into trying to fix something that you just aren't the right person, or there's not the desire from the rest of the organization to fix. And then you kind of have to decide, again, can I live with this, or do I really need to figure out how to fix this? And then how you kind of deal with the challenges that you face is going to kind of define your happiness. It's going to define your career and kind of what you're able to accomplish. And it's going to define your impact on others. So hopefully you can kind of find a way to navigate things and build a career that you're happy with. And that's all I got.
Thank you so much, Dr. Wakefield. That was a great talk. I would now like to open up the floor to a few questions. I think we already have a few in the chat for Dr. Delgado.
I can address Maria's question if vocally, I guess, if that's all right.
I mean, yes, I think looking at a section from the schedule, and I was so afraid that it was going to end at 3, but I'm glad that we actually have some time for questions, and I have so many for both of you. Thank you so much for sharing both of your experiences. It's so kind of refreshing to hear that not everything is picture perfect, because that's what I get fed so much of the time. I don't think people are transparent enough about how they got to such great success. So thank you both for sharing. So for Evan, specifically for you, and then Brian, I also have so many questions for you as well. Some of my questions were, you mentioned that you had this like huge passion to stay in academia, and I think a lot of us in the audience also share that, or a lot of people want to go into industry, etc. But what advice do you have for students who are either close to defending or preparing to start looking for those kinds of opportunities? And then another question is, what did you look for in your postdoctoral position? You mentioned that you did 2. One of them maybe wasn't exactly what you were expecting. So what would you have done maybe differently, conversations that you might have had to either maybe avoid those kinds of issues in the future that you obviously didn't know at the time, but would you have done something differently?
Yeah, I think those are all great and relevant questions. I'm going to fall back on the, cliche that I wouldn't do anything different than what I did, because if I did something different, then I wouldn't be where I'm at right now. So, but that doesn't mean that it was enjoyable, right? So, When I was in my first position, I feel like, like I had mentioned, there were some personal anecdotes that went into a lot of decisions that I made, right? So I was looking for a postdoc that supported me, that I felt like that would really push me forward and I can really be independent. Um, and I found that. It, the problem was I felt like I didn't do enough research on my own to see how the two postdoctoral advisors communicate and interact with each other. If I maybe sat in more lab meetings before I even committed to joining their group, I probably would have sensed that or seen that a little bit like more, obviously. Um, so if you ever have the experience to kind of like almost shadow, I guess, a group, I would take that. I would take that opportunity. If someone invites you down for an interview and then you have the opportunity to just see the daily workings of a lab and a group, I would do that. Or if it's somebody that you're already familiar with, go there for maybe a week, shadow them, like work on techniques that you don't know, but they specialize in, and see how well the training is there. That's really going to determine how successful you are, is the training, really, and the supportive environment that's around you. Um, the personal anecdote was, um, as I graduated, as I defended my thesis, my wife just got into her master's program at Pitt. So and I mean, we were dating at the time, but at that time, I just like, I'm gonna leave Pitt for a postdoc, and then like my wife is just coming here. Like that's a little ridiculous. So I, was invested in trying to make everything in my life work, as well as make sure that it dovetails with my personal and my professional experience too. Um, I believe did I answer your question?
Yeah, you answered both of the questions. I think it's just it's very complicated because everyone's is going to be different. Yeah.
Yeah,
But thank you so much again. Yeah. I'll let the moderators look at the chat and other people ask questions, but I definitely have more.
sure.
Yes, we actually have a question in the chat from Vic Meadows. She says, Dr. Delgado, Dr. Wakefield, can you reflect on how external support and community play into your career paths?
Well, I kind of answered, but a big part of it is my wife has been very supportive of me. We have a very unique kind of relationship uh, regarding our careers. She was a medical student, so she started med school at Penn State, and then I got my position in Delaware, and that was still a 3-hour drive. And then I moved to South Carolina, and that was a 12-hour drive. And then she got her residency in Charleston, which, as established, is a 2-hour drive. So we'd been married for 11 years, and I think we've lived together for 7. And we've not been in the same state for a large portion of our relationship. We met in 2008, and basically we bounced around from basically 2010 to 2018, not being in the same place. So, like having her in her being willing to kind of, we always say we make like a team decision. What's best for the team? It's like it's best for the team right now for you to be in med school and me to be here, but eventually we're going to get back together. And so that was always helpful. And having her there to like, help me deal with some of the crazier things that I talked about. And as I mentioned, if you have really good relationship with your like lab mates and your colleagues, that really helps as well. Having like that good support system professionally, having someone that you can go to and say like, oh, the boss is being terrible today, or like, oh my gosh, I can't believe this student did this. How do you think I should handle it? Like, is really helpful to kind of get you through that stuff. Sort of to cultivate those relationships is really important.
Yeah, I mean, I have to reiterate what Brian said. I mean, my wife is a critical instrument in my success. It's kind of funny how you take for granted familial support sometimes, and they really just help you disconnect from, all the failures that, haunt all of us. You know, it's what I am also pretty terrible at is putting myself out there and solidifying professional support from external individuals. So, for example, having external mentors that are outside of your university or outside of your, space, like that, I think is critical. And I'm learning to even now in my position, like I'm trying to force myself to lean on the people that say, "Come to me, my door is open." Like I just talk to me. And like when people say that, do it. Like don't feel like you have to, you know, have this guard up all the time, that you have to be polished and put together. And I feel like that's the most critical thing in terms of any type of support building infrastructure around yourself and being successful in your career. That's kind of my reflection.
Do we have any more questions from the audience? We have a comment in the chat from a Catherine saying, "That's so reassuring to hear that you and your wife made the distance work for both of your careers. I'm in a slightly similar situation, military spouse, and it helps knowing I'm not the only person having to deal with that." I think Sarah has a question.
Thanks, guys, for the amazing talks and hearing these crazy stories. I was wondering if you could maybe both comment on, it sounds like in both of your careers you had to have some difficult decisions and looking inward and deciding to pivot or move situations, move careers and things like that. I was wondering if you could comment on some of the conversations that you had to have to pivot and how to kind of approach deciding and, talking to the people and letting them know that you are deciding to pivot or move on.
Do you want to go first, Brian, or should I?
You can go first.
All right. Yeah, so that's very poignant. From transitioning from my first postdoctoral position to working in Andy's lab, it was like it was easy, but not, right? So like you never want to have those awkward conversations because who wants to? But the decision, I tell everybody this, and forgive me, whoever's heard this already from me, but I'm always about path of least resistance. So, um, when they were deciding to rehire me, because postdocs in Pitt are on like a annual contract renewal, I was coming up on that annual renewal period, and they were only going to continue my contract for 8 months. And I was like, that's weird. That's really weird. It's like I asked them, I was like, are you going to put me onto a different project? Is that why? And they're like, well, we'll reevaluate when the paper is, ready to be completed. And I was like, okay, so that's that's my answer right there. So, like I said, it was easy for me to already make the decision, like I can't stay here because of the difficult environment that already was there, as well as professionally, like it would be, suicide. So I was looking into like different postdoctoral positions, and I ran into Andy at a seminar, and he said that he asked me how things are going, and I told him about certain things that were happening, and he was like, well, I am hiring a postdoc. Like, why don't you come down and take a look at the lab and we can talk? So I did. Um, now, when I needed to transition from research assistant professor or from postdoc to research assistant professor, that was now having a difficult conversation with Andy. So there, I had to kind of vocalize my intent on staying in academia. Like I knew this is what I wanted to do. I worked this long, almost 10 years in doing this, that it's not like I wanted to give up or whatever, but I knew I could do this if I just got that one bit of funding. I knew I could do it. And I told him that, right now I'm kneecapped at Pitt because all intramural awards are all like you can't apply as a postdoc. You have to have some sort of faculty status. And I needed a research assistant professor position. like to get that type of faculty status so I can apply for junior faculty awards, so I can apply for even R1 awards and things like that. And unfortunately from him, like he couldn't give that to me. He couldn't promise me that he could make the inner workings happen that I can get promoted to research assistant professor. And that wasn't him saying, no, you're not going to get that kind of thing. It was just the logistics behind promoting me in that. And it just so happened that once I published my paper with Andy, that Paul had also opened up a position in his lab for a junior faculty position. So and that's when the conversation happened there. So it's just, I think having those difficult conversations have to happen, obviously, for you to not be stuck. You have to look out for yourself. And it's not that, unless you're in a situation where somebody's deliberately pinning you down and pigeonholing you into like one position, it's usually there's something holding someone else back from, giving you what you think that you need. And if there's something that you think that you need, go and get it. Like if that's my advice.
I don't know if I so much had hard conversations with other people, but it may be kind of hard conversations with myself, kind of realizing when a particular position may have run its course. So kind of with getting what you need. So in Delaware, I kind of didn't feel like I had the instrumentation that I needed. And so I went to South Carolina. Instrumentation wasn't there either. That's another story. But over my time there, it kind of got to a point where I felt like I had built and done everything that I could, but I kind of like reached the ceiling of what was kind of provided to me and the support I could get for kind of running the program I was running at the time. And so it was kind of where's someplace I can go that maybe allows me to do more of this thing that I'm doing, and kind of recognizing that I don't want to be in this environment anymore. For me, the hardest part of all of these moves has been leaving students and colleagues. So that is something that I really struggled with. But just kind of like realizing when a given position has reached giving you all it can give you. And like it's like, hey, is this the right time? And kind of making that decision for yourself.
I think it's about time for our next session. So I just want to thank Dr. Delgado and Dr. Wakefield for their wonderful talks. And thank you so much for sharing your experiences with us. With that being said, I'm going to start off the next session and hand this over to my co-moderator, Cole, who's going to introduce our first speaker.
Hello, everyone. Good afternoon. Like Anna said, we're going to begin session 7, in which we'll talk about inflammation and injury. The first person to start us off for session 7 is Erin Chard, and I will turn it over to her now.
Sorry, can y'all hear me?
Yes.
All right. So I'm going to start off by talking a little bit about a pathogen called Clostridioides difficile. I actually cannot see my PowerPoint on here. There we go. All right. So as you probably already know, the human gut is colonized by a complex and diverse community of microorganisms known as the gut microbiota. But when a person takes antibiotics, it tends to kill off the friendly commensal bacteria in your gut. This leaves an open niche for opportunistic pathogens to colonize, and one of these pathogens that we're extremely interested in is Clostridioides difficile, or C. diff. One characteristic of C. diff is its ability to produce toxins. These toxins can interact with the gut and they induce inflammation and immune cell infiltration. This is what causes the classic symptoms of C. diff infection, including unremitted diarrhea. It can also cause pseudomembranous colitis, and if the symptoms are severe, it can lead to an extension of the colon called toxic megacolon. According to the CDC, C. diff causes about 500,000 infections per year, with 29,000 deaths and an estimated $4.8 billion in excess medical costs. Our lab has previously shown that C. diff colonizes the mucus layer of the gut, but the responses on the host side remain unclear. The mucus layer of the gut is made up of mucins, which are heavily glycosylated proteins, and these can either be secreted by goblet cells, but there are also adherent mucins that sit on the apical membrane of other cell types, including enterocytes. These adherent mucins have many functions, including serving as a decoy for pathogens, intracellular signaling, and wound healing and migration. When we look at Cellex gene, we can see that MUC13 is one of the most expressed adherent mucins in both human and animal colon epithelial cells. So we wanted to take a closer look at what is happening to these adherent mucins during C. diff infection. In order to study C. diff infection, we have a mouse model in which we gave antibiotics in the drinking water, followed by an IP injection of clindamycin and an oral gavage of either PBS as a vehicle control or C. difficil R20291. We then took colonic tissue at days 0, 3, 4, 5, 8, and 10 to look at gene expression via RNA-seq. To monitor disease progression in these mice, we took their weight every day and calculated % weight loss compared to day 0. And as expected, C. diff mice lost weight. And for this experiment, we classified day 3 as peak infection because this was the peak of weight loss. So when we looked at some characteristic adherent mucins, such as MUC13, we saw that with the PBS control mice, there was a steady baseline level of expression. But in the C. diff mice, there was a significant increase in MUC13 at all points of infection. This leads us to think that MUC13 plays a major role in disease recovery. We next wanted to see if C. diff mice had increased protein expression of MUC13 by immunostaining of day 3 colon tissue. And with this, you can see that increase in MUC13 in purple in the C. diff mice at day 3. And this increase was significant. So when we looked into the literature, we saw that many adherent mucins are under control of an anti-inflammatory cytokine called IL-22. And this cytokine can interact with epithelial cells and lead to an increase in adherent mucins. And then when we looked back at the same RNA-seq data from before, we can see a significant increase in IL-22 gene expression at day 3 of infection. So we first wanted to know if C. diff toxins or IL-22 could affect MUC13 expression on in vitro cell lines. And to study this, we treated mouse colon epithelial cells, or CMT93 cells, with different concentrations of IL-22 and with toxins from C. diff. And then from this data, we can see that MUC13 is significantly increased when cells were treated with IL-22 in a dose-dependent manner, but not with C. diff toxin. So this indicates that the IL-22 is being produced from another source to cause the upregulation. We saw the same results in a human cell line, T84 cells, where IL-22 is leading to an increased expression of MUC13. So one of our favorite in vitro models in the INVIC labs are organoids. And this model helps us to understand native tissue in the epithelium. To get these, we first isolate crypts from colonic tissue, and then we plate them in a 3D matrigel dome. These crypts contain stem cells that we can grow in expansion media and then use a differentiation media to drive organoids to differentiate into different cell types of the colon. And when differentiated, you can see this star-like shape under bright field microscopy. And then with IF staining, you can really appreciate the differentiated cells with the goblet cells in blue and enterocytes in yellow. When we treated mouse colon organoids with IL-22, we did see an increase in MUC13 similar to these cell lines. And then we also saw the same increase in human organoids. We next wanted to visualize this upregulation of MUC13 on the protein level through IF staining. And here you can appreciate the visualization of that upregulation. So far we have shown that IL-22, which is characteristically produced by immune cells, is able to interact with epithelial cells, and this in turn causes the upregulation of adherent mucin MUC13. We next wanted to see what cells were producing this IL-22 and what is causing them to produce it. So again, looking through the literature and some data from CellXgene, we can see that group 3 innate lymphoid cells, or ILC3s, and then T helper 17 cells, Th17, are the primary producers of IL-22 in the colon. So type 3 innate lymphoid cells, or ILC3s, were actually pretty recently discovered in the early 2010s. And they are a first-line sentinel at mucosal surfaces where they contribute to the homeostatic and immune response in a major way. They've also been increasingly appreciated as important modulators of chronic inflammation and autoimmune responses. And one of the important functions of these cells is to respond to signals such as cytokines and secrete IL-22. So we actually have a mouse ILC3 cell line called MINC3 cells that we used for a majority of these experiments with ILC3s. And we can use this model to look at IL-22 production in response to cytokines like IL-1 beta or to C. diff toxins. We also wanted to see if these cells produce IL-22 in either the absence or presence of IL-23. So we treated some cells with cytokines that had been indicated as positive stimulators of ILC3s, and we saw that without IL-23 priming, there is no increase in IL-22 production, but when primed with IL-23 and stimulated with IL-1 beta, there was a robust increase in IL-22 production seen in the red. We next wanted to explore whether ILC3s are stimulated by C. diff toxins in the presence or absence of IL-23, but we did not see any increase in IL-22 production with direct interaction with C. diff toxins. So reviewing again what we've done so far, we've seen that ILC3s produce IL-22 when stimulated by cytokines like IL-23 and IL-1 beta. They, however, do not respond to direct stimulation with C. diff toxins. This IL-22 can then interact with epithelial cells and cause an upregulation of adherent mucin MUC13. Since we know that IL-1 beta can cause ILC3s to produce IL-22, I next wanted to look back at the RNA-seq data to see if IL-1 beta was upregulated during C. diff infection. And we do, in fact, see a large increase in IL-1 beta at day 3 post-infection. So since IL-1 beta was so elevated in C. diff infection, we looked into what cells produce this cytokine and saw that macrophages have been largely indicated as IL-1 beta sources in the colon. So upon interaction with the stimulant, macrophages can actually polarize into different types depending on the type of immune response they want to elicit. So M1 macrophages typically produce inflammatory cytokines such as IL-1 beta, IL-6, IL-8, and IL-23. M2 macrophages, on the other hand, are used in anti-inflammatory responses and produce things like TGF-beta, IL-13, and IL-10. So for a majority of this talk, we mostly focus on M1 macrophages since we're looking at IL-1 beta production. M1 macrophages actually have a couple of markers that we can look for. They express receptors such as CD80 and CD86, and then they also express iNOS, and as previously discussed, they produce IL-1 beta. So going back again to the RNA-seq data, I wanted to see if these markers were upregulated in C. diff mice, and I did see a significant increase in NOS2, which encodes for iNOS, and then I also saw an increase in CD80 and CD86 in the C. difficile mice. Our next question was whether macrophages can produce IL-1 beta in response to toxin stimulation. So we obtained peritoneal macrophages from mice and we treated them with C. diff toxins and observed IL-1 beta levels using an ELISA. And from this assay, we did see a significant increase in IL-1 beta with the toxin stimulation, as you can see there in the blue. So another thing about C. diff is that it can produce up to 3 different toxins depending on the strain. These 3 toxins are toxin A, toxin B, and binary toxin. And toxin A and B are the most associated with these symptoms of C. diff infection. So we obtained strains of C. diff that produce only one of these 3 toxins each, and then we treated peritoneal macrophages with them to see if we could pinpoint which toxin is responsible for IL-1 beta production. From this, we see an increase in IL-1 beta with the wild type strain that produces all 3 toxins as we saw before. We did not see an increase with toxin A or CDT, but we did, however, see a large increase in IL-1 beta with toxin B stimulation, leading us to believe that toxin B is responsible for the macrophage response. I next wanted to see if the same supernatant from the macrophages above could elicit a response in the ILC3s. So I plated the MINC3 cells and I treated them with the supernatant of the macrophages, and then I observed IL-22 production with an ELISA. And from this data, we can see that the metabolites from macrophages were treated with C. diff toxins do stimulate ILC3s to produce IL-22. So overall, we have shown that C. diff toxins cause macrophages to release IL-1 beta. This IL-1 beta then interacts with ILC3s, leading them to produce IL-22. And then ILC3-derived IL-22 can then interact with epithelial cells to upregulate MUC13. So our working model is that when C. diff colonizes the colon and produces toxins, it leads to inflammation and immune cell infiltration. When the toxins interact with macrophages, they get polarized into M1 macrophages and produce IL-1 beta in response. This IL-1 beta interacts with ILC3s and causes them to release IL-22. And then IL-22 interacts with receptors on epithelial cells and leads to an increase in MUC13 expression. And we do believe this immune cascade is participating in disease recovery by equipping epithelial cells with protective mucins to help fight off infection and repair damage. And with that, I would like to thank everyone from the Mindy Lab, as well as the sister labs and all of our collaborators, and I will take any questions.
That was a great talk, Erin. I have a question to start us off. It's more about the methodology you outlined for forming your organoids. You stated that you guys use Matrigel, but because Matrigel is collected from EHS mouse sarcomas and possibly contains growth factors, or it does contain growth factors, one of which may be IL-22, do you guys use, or have you guys thought about using reduced growth factor Matrigel?
Not that I'm aware of. I haven't specifically thought of that, but I do think with the cell lines, we do see the same results in the production and everything. So, but that's a good point that we should probably look into.
Thank you. See if there's any questions in the chat as well for you. All right. If there are no more questions for Erin, then I will turn it back over to Anna so she can introduce our next speaker.
Great talk, Erin. If there's any more questions, please do feel free to type them in the chat and Erin can respond to them in there. Introducing our next speaker, we have Maggie Largent from the Medical University of South Carolina.
Hello. Let me see if I can figure out how to. There we go. Okay, can I start? Okay, so I'm going to be talking about Clostridium perfringens pathogenesis when it's exposed to different carbohydrates in infant formula. So the first thing I want to discuss, I cannot figure out. Okay, the first thing I want to discuss is the difference between a preterm infant and a full-term infant. A preterm infant is any baby born before 37 weeks of pregnancy. A full-term infant refers to any baby born at or after that 37-week mark. So preterm infants are 10% of the infants in the US, so it's not a small population. One disease that predominantly affects premature babies is necrotizing enterocolitis, which I'm going to refer to as NEC. It affects 2 to 5% of all premature infants, and it causes necrosis in the intestine due to gas bubbles forming in the intestinal wall. And it does have a 30 to 50% mortality rate. There is no one cure for NEC, but there are a few treatments that can improve the prognosis. These treatments include antibiotics, stabilization, and surgery to remove the damaged part of the intestine. However, it does cost the US $1 billion each year to treat. Survivors of NEC often experience neurological deficits, GI issues such as short bowel syndrome due to the intestine being partially removed due to the necrosis, and poor overall growth rates. Some risk factors of NEC include non-modifiable risk factors such as preterm age, low birth weight, pregnancy complications, and others such as congenital disease. Some of the modifiable risk factors include formula feeding, gut microbiota changes such as dysbiosis, and antibiotic usage. So formula is one thing that is associated with a higher risk of NEC. Formula is used to supplement human milk, and it's designed to mimic it as closely as possible. There are a few different types of formula. Usually it's made with the carbohydrate source being lactose derived from cow milk, but it can also be made with maltodextrin or corn syrup solids, which are derived from cornstarch hydrolyzation. So over time, maltodextrin has been rising in formula usage. So out of formulas available in the early 2000s, the majority were lactose-based, whereas there were some that were maltodextrin, corn syrup solid, or combination of the two, but it was primarily lactose. As we move forward into the late 2000s, combination increases, lactose decreases. Early 2010s, we see a continuing of lactose decreasing in popularity. And as we hit 2020, combination and maltodextrin continue to rise and lactose continues to fall in overall usage. So a couple pros and cons. Lactose does present a lower NEC risk compared to maltodextrin-based formulas, but it can cause allergies in some babies, whereas maltodextrin and corn syrup solids are useful in hypoallergenic formulas as they're less likely to elicit a response, they're cheaper, and they have a longer shelf life. So in animal models, maltodextrin formula has been shown to promote NEC by increasing barrier permeability, increasing inflammation, and increasing NEC incidence and severity. However, healthy term neonates and adults do not show these differences between maltodextrin and a controlled diet. This is probably partially due to differences in the gut microbiome. And one of these bacteria present in a lot of NEC infections that we want to discuss further is called Clostridium perfringens. Clostridium perfringens is a pathobiont that presents as a self-limited infection in adults. It's just food poisoning, typically from undercooked meat. But in infants, C. perfringens is associated with an increased risk of NEC and a reduction in, or an increase in the mortality rate as well. So we wanted to know how C. perfringens was using the carbohydrates found in infant formula, whether it preferred lactose or maltodextrin. And we looked at this over 4 strains to confirm that it wasn't a strain-based, they weren't strain-based findings. So we used 3624, 12916, 13124, and CB1. So the first thing we did is we took a chemically defined medium with no carbohydrates, we grew our C. perfringens, and then we added carbohydrates, lactose, corn syrup solids, and maltodextrin. We then plated this on a 96 well plate, and then we read the optical densities over 24 hours using a plate reader. And then we got our growth curves. So here's an example of a C. perfringens growth curve. The lower bar shows low growth with no carbohydrates, and a peak and then a slight falling off of growth, but overall higher growth with carbohydrates. So for our first strain, 3624, with no carbohydrates, we saw very low growth. With lactose, we saw a peak of growth and then a reduction in the sustainability of this growth over the 24 hours. And then maltodextrin and corn syrup solids both had the same peak at 4 hours, but they had higher sustained growth overall. For 12916, this was very similar. There was a bit more of a falling off from the maltodextrin, but there was still higher sustained growth in the maltodextrin and corn syrup solids versus the lactose. Very similar in 13124, and very similar in CB1. So overall, we reasoned that maltodextrin and corn syrup solids do support more sustained growth over 24 hours of C. perfringens. Then we wanted to find if the formula carbohydrates affect C. perfringens' ability to form a biofilm. Biofilms are formed in a specific fashion. It starts with adhesion, where bacteria sticks to a surface. They then begin to form microcolonies by sticking to each other and the surface itself by secreting DNA and polysaccharides and proteins. And then these microcolonies begin to form a biofilm, which coats the surface and can help protect bacteria from harsh conditions, antibiotics, and helps them stay in their environment. So we wanted to look into this with the carbohydrates in infant formula. So we had much the same protocol as the last experiment, where we used chemically defined medium. We added our own carbohydrates. We plated it on a 96-well plate. We put it in the incubator for 24 hours, and afterwards we performed a crystal violet assay to detect how much biofilm was present. So in our first strain, 3624, we had low biofilm for our control. Our lactose had more biofilm, and our maltodextrin and corn syrup solids had a trend upward of biofilm production. 12916 also had this trend upward between lactose, maltodextrin, and corn syrup solids, though the data was a bit further splayed. 13124 there was a little bit more variation, but we did still see a trend upward. And then CB1 also had this trend upward. We also wanted to see if infant formulas, so the complete formula, not just the carbohydrates, would differently impact C. perfringens biofilm. So we completed the same experiment. However, we used formulas instead of the isolated carbohydrates. On 3624, we can see that there is still this trend upward of control to lactose to maltodextrin. 12916, we had a larger splay of data. 13124, we saw this trend upwards again. And then CB1, we saw this trend again. So overall, we reasoned that C. perfringens biofilm is influenced by different formula compositions. And we saw an overall trend upward from control to lactose to maltodextrin formula. The next thing we wanted to look at was if formula carbohydrates affected C. perfringens toxin-producing ability and its ability to damage the host. So what we did is we took our bacteria, we grew them in lactose or maltodextrin, and then we saw their effect on the host cells to see how they were harmed, or if they were harmed. So the first thing we did is we used qPCR to toxinotype our strains. So we found that 3624 and 12916 were the same toxinotype, whereas CB1 only had one toxin present. And we decided to continue our experiments with 13124 as it was positive for pharyngeolysin O, which has been shown to cause more damage in neck in premature babies specifically. So we decided to continue with this strain for further testing. So the way that we did this is we took our C. perfringens 13124, we grew it in lactose, maltodextrin, or corn syrup solids. We then spun down the culture, removed the pellet, and collected the supernatant of the culture, which contained the toxins. We then plated this with T84 cells, which are human colonic cells, and then we completed a Rasazarin assay, which told us which cells were still metabolizing, which cells were alive, and which cells had passed away. So we saw that when we normalized the data to lactose, C. perfringens grown in lactose being 100% viability, we saw a significant decrease in the viability between lactose, maltodextrin, and corn syrup solids. And so we reasoned that intestinal cell viability does decrease when C. perfringens is grown with maltodextrin and corn syrup solids versus when it is grown in lactose. So in summary, we found that lactose supports lower growth and lower biofilm production of C. perfringens when it is grown in lactose. Maltodextrin and corn syrup solids support higher biofilm production and higher growth rates. We also found that C. perfringens grown in lactose produce less toxins, and maltodextrin corn syrup solids create C. perfringens that produce more toxins. And then we wanted to discuss really quickly some clinical implications. So you might just ask, why not just breastfeed these premature babies? Preterm infants are often underweight and require supplemental nutrition formula, even when they are primarily breastfed. So instead of thinking of eliminating formula entirely, it's more important to think about the optimization. And I want to thank the Bindi Ingovic Lab. I especially want to thank Ali Gutierrez, who was my mentor this summer, for everything that she's helped me with, as well as the sister labs and our collaborator, Katie Chetta. Thank you.
Great job, Maggie. I do have one question for you, since we have a few minutes. In your introduction, you talked about how maltodextrin formulas are only problematic in like preterm models, but not term age or adults. Could you speculate as to why that might be?
Sure. So we think it has something to do with the intestinal brush barrier, which has the like final enzymes that break down complex carbohydrates into individual monomers. So a preterm brush barrier is less developed than a full-term brush barrier, and we believe that the preterm brush barrier has doesn't have the correct enzymes to break down these complicated molecules, as maltodextrin corn syrup solids are much longer and much more complicated than lactose, so that maltodextrin corn syrup solid doesn't get fully broken down and continues onto the colon without being fully broken down, where it can continue to feed the C. perfringens that resides in the colon.
Perfect, thank you. We might have a few minutes for one more question, if anybody has one. Okay, so if anybody has any more, feel free to just leave them in the chat, and I will hand this back over to Cole to introduce our next speaker.
Hey, I don't think our next scheduled speaker has arrived yet, so we'll go ahead and move forward with Kayla McGarry, and she'll tell us her work on how she is elucidating the impact of rotavirus infection on intestinal cell response. And so, Kayla, I'll give it over to you.
All right, sorry, can you guys hear me okay?
Kayla, we can't hear you very well.
You can't hear me?
That's better.
Okay. Yes. All right, thank you for that introduction. So diarrheal diseases remain a serious worldwide health concern, and they're one of the leading causes of mortality, especially among young children under 5 years of age. It's responsible for about 1.7 billion cases and over 500,000 deaths. And 55% of these diarrheal diseases are actually caused by rotavirus or enteric or intestinal viruses, predominantly rotavirus. So rotavirus infection causes over 125 million cases and about 200,000 deaths annually. And while rotavirus vaccines are effective, are a thing and effective in high-income countries with low infant mortality, they're less than 50% effective in low-income countries with high infant mortality. And unfortunately, the only viable treatment option for young children with rotavirus infection are oral rehydration solutions. And these unfortunately do not cause a decrease in diarrhea volume. And due to this, there's typically poor compliance with this treatment method. So really understanding the pathophysiology of diarrheal infections is critical for developing new treatments. In order to understand a little bit more about rotavirus infection, it's important to know the small intestine architecture. So the small intestine is made up of villi, which are these finger-like projections which help to increase surface area for absorption. The crypts are located at the base of these villi. The outer layer of cells is the epithelium. And finally, the lumen is the interior space within the small intestine. And rotavirus infection infects the tips of the villi epithelium in the distal small intestine. And from here, it can have far-reaching effects from infected cells to other, sorry, my PowerPoint is not functioning. To other uninfected cells down the epithelium, which ultimately leads to rotavirus-induced diarrhea. And just a little bit more about the different cell types in the small intestine. We have the enterocytes, which make up the majority of the small intestine epithelium, and they're really important for nutrient and water absorption. The enteroendocrine cells help to store and secrete different hormones like serotonin. Goblet cells are involved in mucus secretion and storage. And then tough cells make up less than 1% of the epithelium, and they're a type of sentinel cell that's involved in different immune responses. And then finally, stem cells are very important for cell renewal and proliferation. And in the field of rotavirus, enterocytes and enteroendocrine cells have already been pretty well studied in their effects of rotavirus infection on their responses in populations. So we really wanted to focus more on the goblet cells, tough cells, and stem cells. and specifically how rotavirus infection affects these cell types and responses. So we already know that a shift in the epithelial cell populations can occur with parasitic infections. So we've seen that things like helminths can actually increase goblet cell numbers as well as tuft cell numbers. Bacterial infections have also shown something similar, causing a shift in the epithelium. With C. diff, we've seen a decrease in tuft cell numbers. However, not much research has actually been done on rotavirus infection and how this causes a shift in the epithelial cell populations. So this leads us to our main question: does rotavirus infection alter epithelial cell responses and types in the small intestine? And in order to investigate this, we used a mouse model of pups that were 4 to 6 days of life. We did an oral gavage of lysate for the uninfected pups and then rotavirus for the infected pups. And from here, we observed the effects. Since diarrhea is a hallmark of rotavirus infection, we decided to measure the percentage of diarrhea incidence in the infected pups to determine the course of the infection. And what we saw with the infected pups was that the infection occurred over a period of about 7 days. At 1 day post-infection, there was about a 25% incidence of diarrhea. And then between days 2 and 4, there was closer to 100% incidence of diarrhea with a peak at day 3. And then with the uninfected pups, we did not see any incidence of diarrhea. So using this data, we decided to collect the small intestine tissue at 1 day post-infection at the beginning of the infection period, and then at 3 days post-infection at the peak of the infection period. And the first cell type we wanted to look at were goblet cells. And as a little reminder, goblet cells produce and store mucus, and they're key to forming the protective mucus layer on the epithelium. Mucin 2 or MUC2 is the dominant mucus in the small intestine, and just a little overview about mucus. It's composed of glycoproteins, and the sugar component of these glycoproteins are the glycans. And if you look closely, you can see that the glycans are made up of a couple different types of sugars. And previous experiments have shown that heterologous rotavirus infection in mice, so this was with a rhesus monkey rotavirus infecting mice, this showed a decreased mucus, decreased mucus, in goblet cells at 1 day post-infection. But we really wanted to look at a homologous rotavirus infection using a species-specific rotavirus, so a mouse rotavirus in mice, to see if we saw similar results. And looking at the 1 day, the beginning of infection at 1 day post-infection for the goblet cells, we first stained for MUC2, and you can see in the green in the micrograph, those represent the MUC2 containing goblet cells. With this, we saw that there was a significant decrease in the number of MUC2 positive goblet cells with rotavirus infection. And this is also indicated in the micrograph. There is clearly a decrease in the amount of MUC2 goblet cells present. And this potentially indicates that rotavirus infection causes increase in mucus secretion from goblet cells early on in the infection period. And while we can stain for MUC2 to determine the presence of mucus, we also wanted to see if the structure of the glycans was changing with rotavirus infection as well. So lectins are proteins that bind to glycans, and we're able to look at these. The specific glycans that we'll be looking at are sialic acid, fucose, and GLC-NAC. And then looking at the beginning of the infection period at 1 day post-infection, we first looked at the sialic acid glycans, which are indicated by the purple in the micrograph. And we saw that with rotavirus infection, there was a significant decrease in the sialic acid glycans present with rotavirus infection at 1 day post-infection. And then the next glycan that we looked at was fucose. And again, we see a significant decrease in the amount of fucose glycan present with rotavirus infection. And then finally, when we looked at GLC-NAC, we did again see a significant decrease in the amount of GLC-NAC glycan present. And this is consistent with our MUC2 data as well. So now looking at the peak of the infection period, we once again stained for MUC2, and this time we did not see a significant difference in the number of MUC2 positive goblet cells between the rotavirus infected and uninfected. And then whenever we looked at the sialic acid glycans with SNA stains, and then the GLC-NAC with the WGA stains, we also did not see a significant change between the rotavirus infected and uninfected and the number of glycans and the amount of glycans present. So this is potentially indicating that these cells are sort of returning to a baseline at 3 days post-infection. So what we've seen thus far is that rotavirus infection stimulates mucus secretion, which results in decreases of MUC2 and glycan stores in goblet cells at 1 day post-infection. And this depletion in goblet cells is restored by 3 days post-infection. The next cell type we wanted to look at are the tough cells, which again make up less than 1% of the epithelium. And since they make up such a small portion of the epithelium, we were able to survey the whole of the small intestine to determine the total number of tough cells in the small intestine. We stained for these using DCLK1. You can see the micrograph, the staining of the tough cell indicated in the purple. And at 1 day post-infection, the beginning of the infection period, we did not see any significant difference between the rotavirus infected and uninfected pups in the total number of tough cells. And then we saw something similar with day 3. We did not see any significant difference between the rotavirus infected and the uninfected in the number of tough cells. So rotavirus infection doesn't seem to alter the expression of tough cells. And the last cell type we wanted to look at were stem cells, specifically how rotavirus infection alters proliferation in the small intestine. And as a little reminder, stem cells are located in the intestinal crypts. And this is the site of proliferation to help renew and maintain the epithelium. And in previous experiments in adult mice, rotavirus infection has been shown to increase proliferation. However, we really wanted to look at this in our more pediatric model. So looking at our data for the beginning of the infection period at 1 day post-infection, we stained for Ki67, which as expected, the proliferative cells were concentrated in the crypts of the small intestine epithelium. And with this, we did not see any significant difference between or in the amount of proliferation with the rotavirus infected and uninfected. However, when we looked at 3 day post-infection, we did see that there was a significant increase in the amount of proliferation with rotavirus infection at the peak of the infection period. So in summary, rotavirus infection increases mucus secretion from goblet cells at 1 day post-infection, and this mucus depletion is restored by 3 days post-infection. We also saw that rotavirus infection doesn't seem to alter the expression of tough cells and increases proliferation at 3 days post-infection. I want to thank all of our funding sponsors, as well as the organizers of PISA. I also want to thank Dr. Kristen Ingevik's lab and the other sister labs for everything, and I will now be open to any questions.
That was a great talk, Kayla. I have a question for you. You said that diarrhea is a defining feature of rotavirus infection.
Yes.
If you consider the virus's effects on intestinal glycans, how might rotavirus infection affect or alter microvilli structure and function? Because altered or impacted microvilli has been linked to an increased incidence in diarrhea as well.
I'm not entirely sure how to answer that question. This is a little bit beyond my scope of research. We just know that there is a decrease in the amount of glycans present with rotavirus infection. Yes.
That was great, Kayla. I really liked your whole Swiss roll immunostaining. I've never seen it like that. I was wondering if you were interested in looking at panda cells, or if you did get to look at them or have plans to look at them.
Yeah, that could definitely, we haven't gotten to look at them quite yet, but that could absolutely be an area of further research. Yeah.
I also had another question. So you said that the goblet cells restored by post-infection day 3. Do you have any idea or theories of why that may be?
Yeah, so we think that the rotavirus infects the tips of the villi, right, and causes some sort of signaling down the epithelium. And this kind of, we think it stimulates the release of the mucus from the goblet cells early on in the infection period. However, by day 3, they seem to kind of restore this amount of mucus. They're constantly creating mucus. It's not like it's a continuous process. So they're just always restoring their mucus. But we think that these cells might be kind of returning to a baseline as they adjust to the infection at this point in the infection period. So we'll be investigating a little bit further later in the infection period to see if there's also any other changes in the glycans, or sorry, the mucus stores in the goblet cells as well. So
Thanks.
Okay, well, if there's no other questions for Kayla, you can leave any as you think of them in the chat for her to respond to. I will move on to our next speaker, Celine Shore, also from the Medical University of South Carolina.
Can everyone hear me okay?
Yep.
Awesome. Now I'm gonna share my screen. Just a little bit. Everything look good? Okay, cool. So thank you, Anna, for that introduction. To start with, histamine is an important biogenic amine involved in several physiological processes in the body, including allergic reaction, gastric acid secretion in the stomach, and neurotransmitter activity. It is made by a lot of our own cells. So for example, mast cells will release histamine during an allergic reaction. Enterochromaffin-like cells will also release histamine in the stomach, and our neurons will as well. But there is a third, or an unappreciated member of our cells that can produce histamine, and these are our microbiome. And so what microbes do, particularly bacteria, is they'll bring histidine from the environment into their cells, convert it to histamine using a histidine decarboxylase, and excrete it back out into the environment. And so we were really curious about which bacteria in the gut may be doing this. It's already well understood that several bacteria involved in fermentation and in food processes can do this, but we wanted to know in the human body if this may be happening. So what we did was we leveraged the fact that in order for bacteria to make histamine, they need to have this histidine decarboxylase gene, which has a which can all be categorized under a particular code, which we can then look and query within bacterial genomes online. And so for this, we used the Integrated Microbiome Genome Database and examined over 100,000 genomes of bacteria. We found that almost 4% of all bacteria within this database had this histidine decarboxylase gene. And when we looked at those members, while many of them were terrestrial and aquatic, a good portion of them came from both the human gut and can be found also in the oral cavity. So almost 14%. Majority of these were proteobacteria, but we have a nice representation of other phylum here, and over 100 different genuses are represented as having this gene. So from some that you may be familiar with in your own data, we have Clostridium perfringens, which has already been talked about today in this session. We have Fusobacterium varium and Bacteroides fragilis. And we also have L. reuteri, which is a probiotic in the gut as opposed to a pathogen. And so while we find that both commensal bacteria and pathobionts or pathogens can have the potential to produce histamine, we were curious what this histamine would be doing. And so in order to probe this, we decided to first look at the target of histamine in the gut and oral tract. So histamine has one of 4 targets. It's 4 receptors, 1 through 4. Histamine receptor 1 is primarily associated with allergic responses and allergies in the airway epithelium. HRH2 is found in the stomach and is involved in acid secretion. It's also involved in our immune cells. HRH3 is important in the brain for neuronal signaling. And then HRH4, though not a lot is known about it, is found to be involved in immune modulation. And so before I get into whether our cells in our intestines have HRH1, I just want to remind you guys of the cells that are present. Kayla actually already did a really good job of this. So really briefly, in the small intestine, we have both crypts and villi that are covered in mucus. The majority of the cells are enterocytes, but we also have endoendocrine cells as well as tough cells. Goblet cells, which produce mucus, we talked a lot about that. Tanith cells that are antimicrobial, and then we have our stem cells in the small intestine. And when we move to our large intestine, and of course these are all covered in mucus. But when we move to our large intestine or the colon, it looks similar, but it's not exactly the same. So we only have crypts here, no villi. We have a much thicker layer of mucus. But we do have many of the same cells as found in the small intestine, although we don't have any Paneth cells here. And so in order to probe whether these cells in the small and large intestine have histamine receptors, we went to a single cell sequencing database and probed whether individual cells have these receptors. So as a kind of positive control, we looked at parietal cells in the stomach, which are known to produce histamine receptor 2 and only histamine receptor 2. And we find that that is the case in the single cell sequencing data. When we look in our large intestine, we find that all of the cells mentioned, so goblet cells, endoendocrine cells, stem cells, and enterocytes are expressing histamine receptor 1 and only 1, with the exception of tough cells, which are not expressing HRH1. And then we look in the small intestines. This is very similar, with again our one exception being tough cells that are not expressing HRH1. In this case, they appear to potentially be expressing HRH2, which is really, really interesting. But the majority of these cells are HRH1. And so Erin also actually already introduced our model system, which is a organoid model for the intestine. And so really briefly, we can isolate crypts from different parts of the intestine and expand them in matrigel to create organoids, which we can then differentiate and test on. And so when we look at expression of HRH1 in organoids, so no longer in the single cell sequencing data, but in an actual organoid model, we again find that no matter where we isolate our organoids from, we are only seeing expression of HRH1 in these epithelial cells. And so our next question was whether HRH1 is acting in the gut as it does in the lung epithelium. So airway HRH1 is pro-inflammatory. So in the lungs, histamine will bind to HRH1 and lead to release of calcium, as well as a phosphorylation MAP kinase pathway, which eventually leads to the release of cytokines, which is why it's pro-inflammatory. But is this happening in the intestines? And so we, again, are using our human organoid model, where we are able to transduce our organoids with basically a fluorescent reporter for calcium. And so essentially when calcium is present or is increased, you'll get an increase in this fluorescent signal. And we can use that as a readout for whether his calcium is being released in our cells. And so then we take our organoids and monolators, we can then treat them with either vehicle or histamine, and it should be pretty clear from this video that's hopefully playing for you guys that in the presence of histamine, we get higher levels of fluorescence, which means we're getting high levels of calcium detected in our cells, meaning that histamine through HRH1 is leading to calcium release in these organoids. And we can quantify this. So again, our vehicle control, you see, has low levels of any detected fluorescence. But then what's really nice is that in a dose-dependent manner, the more histamine we add to our cells, the higher fluorescence we get. Again, kind of leading to that idea that histamine signaling is leading to calcium release. And so we've kind of confirmed that this first part of the model of what we know happens in the airway epithelium is also happening in the gut. But we next wanted to know whether we're getting phosphorylation of our MAP kinase pathway or cytokine release. And so essentially, we find that we cannot detect any differences in MAP kinase phosphorylation. So we looked at P38, saw no differences here in the presence of histamine, ERK1 and 2, JUNK, KREB, AKT, or NF-kappa B. We see no difference in phosphorylation in the presence or absence of histamine, suggesting that histamine signaling in these organoids is not activating the same MAP kinase pathway as in the airways. But is it leading to release of pro-inflammatory cytokines? We took a very similar approach as we had previously. But long story short, none of the inflammatory cytokines that we looked at in a very large panel were increased when we added histamine to our organoids. And I will say we had positive controls for all of this. It's just obviously all our data here is pretty negative. And so in conclusion for this part of the talk, histamine is activating calcium signaling in the gut, but it's not activating a MAP kinase pathway or the typical one or releasing inflammatory cytokines. And so what is it doing in the gut? Again, we decided to focus on particular cells here. And one of those cells that was expressing HRH1 that is one of our favorite cells is the goblet cells. And so as was already talked about by Kayla, goblet cells release mucus, primarily MUC2. And so we were wondering whether histamine signaling impacts mucus secretion. And so for this, we moved to a pig explant model where we removed the colon of a pig, separated in half. So these are the same part of the colon, just different halves. We applied our buffer control to one half or our histamine and buffer to the other. And then we were able to collect any mucus that had been released, as well as stain the cells that were left over to see with Alcium blue staining whether the mucus was still inside the cells in these colon cells. And so what we found is that MUC2 was up in what was released from the cells, suggesting that histamine is leading to the release of MUC2 from these colon cells. And when we stain our cells, we do see a difference as well. So on the left is our buffer control, where you can see the goblet cells within our crypts are still full of mucus. They are very nicely contained. Whereas when we add histamine, several of the goblet cells have clearly released mucus into the outer parts of the cell here, which further supports that mucus is being released in response to histamine. And so we finally have one additional way to measure whether mucus has been released from our colon organoids, and that's with a mucus expulsion assay, where we're able to fluorescently tag the mucus in our cells, apply our drug of interest, and collect supernatant to see if the mucus is now outside of our cells. And so we again did this with a buffer control or histamine. And this is just showing what it looks like before we add drug, where you can see in red this time that our cells are containing nice mucus within them. And so what we again find with this assay is that when we add histamine, we get a release of mucus into the supernatants, and very nicely, as histamine concentration goes up, we get more release of mucus. And so this kind of is clearly telling us that HRH1 is involved in colonic mucus secretion, wherein histamine binds to histamine receptor 1, leading to calcium release, and therefore mucus secretion, which is involved in compound exocytosis. And so we were also wondering, though, whether other mucus secreting cells could also be stimulated by histamine. And for this, we were particularly interested in salivary glands in the mouth. They actually stain very similarly to goblet cells, although these are not goblet cells that are in our salivary glands. And so you can see very nicely. I don't have a timer on my screen. I
You
apologize.
can continue talking. We're running forward in this session.
Okay, so
You're
sorry
good.
about that.
Continue talking.
And so we
No,
did
you're
many of
good.
the same assays with salivary glands as we did with the stomach. Single cell sequencing, again, we only see HRH1 expressed in salivary glands. We used an organoid model to measure whether mucus is being released, and we can see there's mucus inside the cells of these organoids. So when we add histamine or buffer, we again see that with increasing levels of histamine, we get an increase in mucus released. And we are also not detecting any changes in inflammatory cytokines from salivary glands with histamine. So our working epithelial model is that bacteria in our gut and in our oral cavity are producing histamine, which can stimulate our mucus-producing cells to release mucus, which is therefore acting in a protective measure. And we were curious about whether this has any impact or whether pathogens have any impact on this process. And I'll make this really brief. So Erin already did a very nice job of introducing C. difficile, one of our favorite pathogens. I will just add that it loves and it does most of its infection inside the mucus layer of the large intestine, and can actually, a way our body protects ourselves is through getting rid of it through mucus release. And so we were first curious about whether C. difficile infection impacts expression of our histamine receptor. We found that it does not. And so we were next curious about whether C. difficile toxin may impact mucus release from histamine stimulation. And in this one, thankfully, well, either way, it does. So you can see on top our normal calcium release in response to histamine. And on the bottom, when we pre-treat our cells with C. difficile toxin, there is a slight increase, but it is inhibited compared to without C. difficile toxin. And when we quantify this, this is actually more obvious in that we only are seeing a significant increase in calcium release when C. difficile toxin is not present. So what does this all mean? This kind of reframes the paradigm that histamine is pro-inflammatory in the intestines. And so what is actually maybe doing in part is that bacteriohistamine may stimulate goblet cells to release mucus, and that pathogens could potentially inhibit this pathway. So thank you so much for allowing me to speak to you today at PISA. I'd like to thank also my funders, my lab, our collaborators, and with that, I'll take any questions.
Great job, Celine. I do have one question. When you were talking about your phosphorylated assays, can you touch a bit on how you did that? Did you do Western blots, or what assay did you use to quantify phosphorylation?
So for this assay, we actually used a company that specializes in detecting phosphorylation of different proteins. And I'm blanking on the name of the company right now, but we were actually able to design our own custom panel of proteins we were interested in measuring for phosphorylation, and able to do that in a more high throughput way than us doing our own Westerns.
All right, I think Maria has a question.
Yes, I have maybe a kind of naive question, but for mostly the first part of your talk, you were talking about the calcium flux, and we know that calcium flux is also coordinated with potassium movement as well within the cell. So I was wondering if you've looked as well as the potassium movement and levels or changes in the membrane potential of the cells, and whether or not that would maybe alter that mucus production that you more so focused on that second half of your presentation.
Yeah, that is a very interesting question. So, yeah, so As far as I'm aware, I'm not, when I think of calcium, potassium, I think more of neurons, but obviously those same pumps are present in other cells in the body. And I'm not aware of what potassium is doing with goblet cells. But in my previous lab, we were interested in how toxin and antitoxins impacted potassium flux in bacteria. And so we could easily apply those same methods in where we can monitor potassium and see what's happening with our cells, or apply our own potassium and try to get at that question.
I have a question for you, Celine.
Yeah.
What mechanisms might explain why histamine signaling through HR1 enhances mucus secretion without activating inflammatory pathways?
Yeah, so in the respiratory field, there's a lot of conflicting data on whether there is a direct connection between release of calcium and the MAP kinase phosphorylation